Biotechnology and Food Technology, University of Johannesburg, South Africa. Dr. Debati Pal, Associate Professor, Amity University, Institute of Biotechnology, Noida, New Delhi. My colleagues, Dr. N. Kishore, Dr. Shantipriya, Dr. Mrudula, and my dear students and other faculty members, research scholars, a very good morning to you all. Today's one-day international webinar on new frontiers in biotechnology research for social and environmental welfare, organized by Department of Microbiology, Palamur University, Mahabubnagar, Telangana. This webinar is planned because we know last 2 years, COVID-19 pandemic has shattered our lives. It not only disturbed the economy, but also it affected academics very badly. And actually, we were not prepared for such a new system where we can interact with the eminent scholars or eminent speakers throughout the globe. But this pandemic has taught us a lesson, and because of that, now we are able to conduct online classes and even interact with eminent personalities throughout the globe, conducting various webinars, various online symposia, workshops, and all. And with regard to our department, Department of Microbiology is established in the year 2008 with the establishment of our university. It was started by our founder, Vice Chancellor, Professor Gopal Reddy Garu, under the guidance of Professor Gopal Reddy Garu, Senior Professor from Usmania University, and myself, along with my colleagues, our senior most Professor Pindi Pawan Kumar Garu, who is Chairman Board of Studies and Registrar of this university. My friend and colleague, Dr. N. Kishore, who is head of the Department of Microbiology, and my colleague, Dr. Shantipriya, Dr. Suma Mukherjee, and we have got Dr. Shankar at PG Center Vanaparthi, and Sanjeev Kumar at Vanaparthi as faculty members. And we have got more than 138 student strength with 17 research scholars at our department pursuing PhD. We are very happy that after establishment of department, we have conducted many webinars and many seminars offline and online. And even during pandemic, we have conducted more than 10 seminars, actually, international and national seminars. Here, one important thing what we'd like to say is our present Vice Chancellor, Professor L.B. Lakshmikant Rathod Garu, because it is almost 10 months after he has taken charge as Vice Chancellor over here. From the day one, he is behind all the departments. He is asking all the departments to conduct various research activities. And not only that, he is encouraging research by providing seed fund for every faculty for attending seminars, for writing for projects. And because of that, today we are able to, conduct many seminars after Sir has taken charge. And even this academic year, Sir has planned to construct, a common research facilities building worth of 10 to 15 crores. Really, it's a very good initiative to our university where we can carry out our research. And with regard to this webinar, we all know biotechnology has, wide aspects, wide applications where during pandemic also we have seen development of various immune booster related diets, drugs, and even biotechnology has given us various solutions for rapid diagnosis of COVID situations, and even it has helped us for drug repurposing and even designing various recombinant vaccines. So keeping all this in a way, we have planned this webinar where actually we have got 3 eminent personalities where they will stress on. First, Dr. Suma Mukherjee. We welcome you, ma'am, where she will be addressing enhancement and encapsulation of nutrient efficacy in the food systems. So that can play a major role in boosting our immune responses, which is a major shield during pandemics, what we have observed. And we have got Dr. Shizwe, Dr. Shizwe, where he will be taking finding new drugs from silent biosynthetic. I'm sorry. Yeah, he will be taking a topic: functional characterization of biosynthetic gene clusters in marine-derived microorganisms, where many times we know till now we say that maybe 1% or less than 1% has been tapped with regard to microbial sources, and major sources is confined to marine environments. And tapping such sources can definitely produce a wonderful drugs or wonderful food related particles. And we have got the third speaker, Dr. Debati Paul, Associate Professor, Amity University, where she will be dealing with polygenous red yeast strains for single cell oil and carotenoid production, a waste to value approach, which is the need of the hour. So without taking much time, I request my colleague, Dr. N. Kishore, to introduce the first speaker. Thank you, Ananda. Yeah. I'm sorry. So before that, I request Dr. Noorjahan, Principal, University PG College, to address the gathering. Ma'am, please join me.
I'm audible?
Yeah, ma'am, please.
Yeah, thank you for inviting me to this international webinar. First of all, I would like to thank Professor Lakshmikant Rathod Garu, Honorable Vice Chancellor of Palamur University, and respected Registrar, Professor Pindi Pawan Kumar Sir, and BUS Chairperson, Department of Microbiology, and the conveners of this international webinar, Dr. Madhusudan Reddy, Director IQAC, and Dr. N. Kishore, Head Department of Microbiology, and co-convener Dr. Shanti Priya, and all the other organizing committee members, Dr. Mudula, and the research scholars, and the various faculty members from the PG centers, and my dear students, a warm welcome to all. On the outset, I would like to congratulate the department faculty to take who has taken the initiative for organizing apt title for discussion on this day, because we have seen, as already the convener has introduced to you, what are the situation prevailed since 2 years, still the crisis is not yet over, and how this biotechnology has put live examples to overcome this crisis. We have seen the COVID crisis, and during COVID crisis, where we are new to this crisis and faced a lot of problems in the healthcare system, and the biotechnology and the genomic medicine, which has introduced mRNA technology and introduced the vaccines, and where we could overcome and less effect on the human systems can be seen, which are the live examples of the biotechnology. I would like, because I'm from the chemistry background, I will broadly classify the biotechnology and its applications on 3 categories, like the healthcare system and the industrial or the economical side and the food. So where the people have the opinion that we are very optimistic towards the recent trends, what is going on in biotechnological research for the medicine. And we are encouraging the new technologies in the biotechnological research, which can help us to overcome the various new vaccine viruses or the new crisis like pandemic situation what we have faced, and that can also show some light on the non-communicable diseases like diabetes and hypertension also. So we are hoping new technologies can be invented in this areas also. But whereas for the industries, a moderate support is given for the biotechnological research, where the biofuels and the changes in the industrial products are welcomed. But when come to the food technologies, we are not encouraging the biotechnological and the GM products. And there is a adverse opposition is seen in the GM food. So let us see how this discussions can show some light on these 3 areas, and that can overcome our misunderstandings in the area of biotechnology, and let us encourage the new research scholars to take up some challenges in this area. So I thank all the organizers for organizing such a wonderful seminar on this topic, and I take this opportunity to thank all the faculty members of the Department of Microbiology for giving me this opportunity. Thank you.
Thank you, ma'am. Thanks for your brief address and encouragement. Thank you. Now I request our respected registrar sir to address the gathering. So please join. Hello.
Thank you, Madhu. Thank you so much.
Yeah, please.
Honorable Vice Chancellor of this university, Professor L.B. Lakshmikant Rathod garu, and today's respected speakers, Suma Mukherjee, University of Holy Cross, USA, and another speaker, Dr. Sizwe, University of Johannesburg, South Africa, and third speaker, Dr. Debarati Paul, Amity Institute of Biotechnology, Noida, and our respected principal madam, Dr. Nurjahan, and conveners of this webinar, Dr. D. Masudan Reddy, who is due to Vice Chancellor and Director IPSC, and Dr. Kishore, Additional Academic Director, Palam University, and Dr. Shantipriya Madam, Additional Controller, Palam University, and all other faculty members, and my dear student friends, and other faculty members, distinguished guests, a very good morning to you all. So today, this important topic, where we are talking about the importance of biotechnology. Biotechnology today is a sunrise area with potential to meet the basic human requirements of the human being. Societies have a very important role to play in the promotion, awareness, and sensitization of science and technology. Programs and related activities, they have become an integral part of the application of science and technology for societal welfare. And the recent developments in the field of biotechnology and its allied subjects shows the interest of scientific community towards innovations, innovative products for the benefit of human beings and environmental concern. Biotechnology is a field which has developed all over the world with phenomenal speed. Its interventions and relevant for improvements in the area of agriculture, healthcare, environment, industry, and employment generating activities. We have seen in last 2 years the pandemic situation. Everybody was the panic due to corona, and even what we are seeing nowadays, the children are becoming very susceptible for manageable infections. On the other hand, the organisms are acquiring a resistance pertaining to the general principle, the survival of fitness. This often leads to the genetic mutations causing new diseases. Nutrition in food is depleting. Sperm count in male is failing drastically. Fertility now has become something which can be acquired from a laboratory. A separate segment in medical science has been added in the name of lifestyle disease. For everything becoming, we can see that the linked to the environment, and for every action against the environment, you will be the witness. The consequence may not be overnight, sometimes generations together. So we cannot blame or attribute to mistakes to geography, community, or generation, as everything on earth reciprocates to all human in one or other way. The combination of different methods is still the most suitable way of having a better understanding about diversity, phylogenesis, ecology, evolution, and taxonomy of the largest group of living organisms on the earth. Research using novel and efficient tools to identify and characterize all the living communities has been the key of elucidating biological activities with potential for industrial applications. Metagenomics, the culture-independent cloning and analysis of microbial DNA extracted directly from an environmental samples, functional or sequence-based screening or metagenomic DNA libraries are providing useful for exploitation of untapped microbes. Environmental genome sequencing efforts are aiding better understanding of the community structure and their phylogenetic importance in the ecosystem. Scientific perceptions of the microbial world and its extreme fringes have changed with the increasing pace and with unforeseen consequences and lead directly towards the grand research challenges in the future. The advances in molecular biology and development of new tools and technologies have created unprecedented opportunities for crosstalk amongst the subdisciplines of microbiology and allied biological sciences. Hence, it is important to catalyze and facilitate formation of multi-subdisciplinary networks based on needs and emerging challenges of human and animal health, agriculture, environment, energy, biotechnology industry, etc. The topic chosen for this conference is very relevant to meet the present need of the country. Such conference provides opportunities to exchange new information and learn the latest research in the field of biotechnology. To address the complex issues faced by the modern society, the scientists and engineers must have unique capabilities. This potential in our young researchers will probe and explore problems to find the critically evaluate information and to effectively communicate our research findings to others. These are extremely useful for young researchers and students as it provides a platform to meet experts and discuss their research and seek guidance. In this context, the webinar will provide a wonderful platform for all the participants, and they can refresh and strengthen their knowledge and explore the most updated information regarding the applications of biotechnology. The conference will provide ample opportunities for all of the upcoming generation. They can interact with the leading scientists and experts in the field of biotechnology and its interdisciplinary areas. The plenary talks from top-level scientists, leading experts will provide you the latest information in the field of biotechnology. I take this opportunity and congratulate our colleagues, friends for conducting this wonderful event, and I wish all the success of this webinar a resounding success. Thank you, Anand.
Thank you, sir. So you have touched all the topics of today's webinar, and really wonderful about how you said about role of microorganisms affecting the human life with respect to pandemic, especially local environment, and how they are helping the microorganisms by mutations and how to survive in the present situation, and novel research tools to address the problems, and especially advancement of life sciences is the key for many solutions. So with this, really, I thank you, sir.
Thank you.
And I welcome our Honorable Vice Chancellor, sir, to address the gathering, sir. So please join.
Okay, I'm audible?
Yes, sir. Please, sir. Please.
Okay. Very good morning to you all. Respected Registrar of Palamur University, Professor Pawan Kumar Garu, and our senior most faculty, Professor Gopal Reddy, former Head Department of Microbiology, Usmania University, Dr. Suma Mukherjee, University of Holy Cross, Los Angeles, United States of America, Dr. Switch Nodelova, University of Johannesburg, South Africa, Dr. Deborah all, Amity Institute of Biotechnology, India, and my colleagues, today's all the faculty members. I have great pleasure to be with you this morning for the inauguration of the international webinar on new frontier in biotechnology research for social and environmental welfare. At the very outset, let me deeply appreciate the initiation of our Department of Microbiology. The international webinar for this webinar, the convener of this webinar, Dr. D. Masudar Reddy Garu, and my another colleague, Dr. N. Kishore Garu, co-convener, Dr. Shantapriya Azmira Garu, and Dr. M.S.R. Anjanelu, and their organizing team members for organizing this international webinar in collaboration with the Internal Quality Assurance Cell, Palamur University. It is indeed a befitting that eminent academician from United States of America, South Africa, and India are gracing this webinar as initiated speakers to enlighten us on the focal theme. There are more than 100 participants in this international webinar. I strongly believe that such inter-institutional collaboration holds the key for promotion of academic excellency in higher education. Needless to say, scholarly and scientific events like this go a long way in expanding the avenues of knowledge and providing intellectual space for exchanging of ideas and experiences, sharing to our faculty, research scholars, and students. The theme you have chosen for the webinar is of typical significance and societal relevance. This pandemic 2019, the microbiology department has key role in this pandemic. I am of with view that biotechnology as an existing field of knowledge and successfully built strong bridge between different realm of life science and ever expanding domain of technology. In doing so, it has emerged at the center stage of knowledge, economy and society in 21st century. Biotechnology marks a paradigm shift in interdisciplinary research pursued for human welfare. It has visible impact on all facts of human life, be healthy, environmental, or agriculture. I am sure listening to the distinguished top speakers from different parts of the world in this particular theme and participants in the immersive academic event will be meaningful learning experience for our academic fraternity, and the keynote speakers, they will deliver their good academic themes. This is useful for our research scholars and students. Once again, I am very much thankful to the organizers. They have organized this international webinar in a very good manner. Thank you, one and all.
Thank you for blessing us, sir, with your kind words. Really, you said about the need of webinars in exchanging knowledge and need for interdisciplinary research. We have initiated in our Palamur University also, because you have taken initiation where we are in collaboration with the Department of Chemistry, Zoology, Botany, and even sometimes with the Economics and Commerce and Business Management. We are planning various programs at our university. And from IQAC part also, you have asked us to initiate many research programs in our university. And really, we are very thankful and blessed to have such a vice chancellor with a good vision. Thank you, sir. Thanks for joining us.
Welcome.
So now I take a pleasure in asking my colleague, Dr. N. Kishore, one of the conveners of this webinar, to introduce the first speaker. And after the first speaker, I request all the participants to post questions in the chat box so that your questions will be addressed by our research scholars, where the concerned eminent speakers will be answering those questions. So now I request Dr. N. Kishore.
Good morning, one and all. I'm happy to introduce our first speaker, Dr. Suma Mukherjee. Dr. Suma Mukherjee is an assistant professor in the University of Holy Cross, United States. She started her career from West Bengal, India, where she has completed her undergraduate and master's program from North Bengal University in Botany. She completed her first PhD from Vishwabharati Central University from India. However, she pursued her career in food science in United States, where she completed another PhD from Mississippi State University in food system and functionality. She worked as a postdoctoral associate in 2 universities under 2 National Science Foundation projects and several multidisciplinary national and international projects. She has more than 12 research publications, 9 book chapters, 18 conference publications. She mentored more than 350 students in her short teaching career. She is awarded with endowed professorship grant and another grant from Louisiana Board of Regents in 2021 from her institute. She is also a leading research coordinator for undergraduate summer interns funded by Brown Foundation of State of Louisiana, United States. I would now like to take the privilege of inviting Dr. Suma Mukherjee to deliver her talk and enlighten us. Over to Dr. Suma Mukherjee.
Thank you. Good morning, everyone. Respected Vice Chancellor, Professor Rathore, respected Registrar of the Palaburu University, Dr. Kumar, respected Principal and Chairperson, Dr. Nurjahan, conveners, Dr. Reddy, Dr. Kishore, Dr. Ajmi, and respected members of the organizing committee, faculties of the Department of Microbiology, and all the respected faculties present in this webinar, our research scholars, and the students. First of all, I would like to thank you all for inviting me to talk here. Then I would like to introduce myself a little bit. I am Dr. Suma Mukherjee. So as Dr. Kishore said, talked about me, that I started my career in the West Bengal. And so I will I will share my slides so that students and all speakers can follow. So I'm really honored to talk here. So as Dr. Kishore said, I work for the University of Holy Cross, and I started this position in 2020 at the middle of the COVID situation. And let's talk about a little bit of my career first. So I finished my BS and MS from North Bengal University, West Bengal in the year 95 to 2001. And my first PhD I finished from Vishwabharati Central University, India, in 2009. After that, I moved to US. So life is always being challenging for all of us. So life was challenging when I first came to United States. So for the need of my pursuing career, I pursued another PhD in food science from the Mississippi State University. And I finished that PhD in 2017. And after that, I pursued 2 postdoc positions. So one position was completely different from my field of research, because that was in horse asthma research. I pursued 1 year. But the knowledge I have gathered in different fields as I jumped from botany to health science, food science, I have gathered huge knowledge that I can today apply in the field of I'm working. So I want to tell my students and all the audience here, the knowledge we gather doesn't go from us. It remains with us and it makes us strong to fight this life. So let's see my professional positions. So I'm coming from the bottom of my slide. So I was a research associate in the North Bengal Agriculture University for 2 years. I used to work with CIMIT organization, and from 2012 to 17, I was a teaching assistant and instructor of record for the University of Mississippi State University, where I was taught how to teach in the university condition. And then postdoc in Mississippi State University, and another postdoc in Arkansas, University of Arkansas, where I worked with a very eminent scientist in my field of research, Dr. Namo Emulsion. So she taught me so many things that today I'm here. And after that, 2020, I started my career. So let's come back to our slides. So what main talk we're going to talk. So a few slides, I will give you some warm up for this good morning. So what we eat, my area of research is food science and food system functionality. So what we eat, major constituent of food, two parts we divide. One is macronutrient and there is micronutrient. So among micronutrient, we eat carbohydrate, proteins and fats. And micronutrient, we are eating like water-soluble vitamins and fat-soluble vitamins, trace minerals and microminerals. So now this next slide. So what do we eat? There's a huge difference between Western diet and Eastern diet. So I have given two sides in the left-hand side, all food items for breakfast consumed by Western people. So there are bagels, pancakes, scrambled egg, and different other food items. There's a sweet thing, coffee, and other stuff. And in the Eastern diet, whereas we eat like upma, vara, or this kind of like roti, and some rice, flatbread, etc. we eat. So now my concern, also like lunch and dinner concept, like Western diet, they eat like fried chicken, fried meat, and preserved meat like pancetta and this type, that is this type of meat, and burger, fries a lot. And for Eastern diet, always we eat like home cooked meat, like rice, chapati, and this type. So our lunch and dinner are almost like same for Eastern people and also Western people. It varies person to person. Now my concern, my thing, like what the main concern in Western diet. So in this Western diet, protein content is very high, sugar content is very high, saturated fat content is high, and less phytochemicals, less fiber, high calorie, high amount of preservative are there because they consume all this like canned food and packed food a lot. And for the Eastern diet, our protein content is less. Carbohydrate content is very high. And the third point, like food distribution among population is not adequate. So this is not our area of research, but this is about the economic condition of India. And fourth point is nutrient content in food is less due to the cooking pattern. So that the way we cook food, we lose some of the very like important microminerals and vitamins, we lose. So next comes, as a food scientist, when we work, first thing we see the market, how we are going to make products and how we are going to make money. So in Western market, you will see the number of products in any food market is like humongous. Like it's a huge number of food products that are available in the market. And whereas in Eastern market, still like we consume vegetables, fruits, those are fresh and home cooked. And because of the globalization of these days, like shopping malls and stuff, and like we are getting all food all over the world. So now people are also getting like packed food and stuff like, so like this pattern is changing in India too, but still people eat more homemade food. Now about the amount of food we produce. So in India, we need to feed for today's population like 1,403 million people in India. We need to food. So the amount of food we need to produce to feed this many people, whereas in US this is less, 332 million people we are feeding. So there is a difference, like the amount of food we need to produce in India and the amount of food we need to produce, there is a difference here. Now, the global scenario of the food market. So we are thriving on more ready-to-eat food product these days. And if we see the market, like every minute a new product is launched in the market, and number of products are increasing due to globalization. The market of food ingredient is rising sky high. So what in this fourth point, what I mean to say that like only the food, like one kind of flour or two kinds of flour were available like 20 years before. But if you now go to the grocery store and you can get like 20 to 25 different types of flour are available for making your chapati. So this is like food ingredients, number of like the food ingredients we are making high, and we are giving people choices so that they can select what they want. Oatmeal, from oatmeal to different kind of flour, different kind of whole grain, we are adding to our diet. And last point is the major concern also for environmental and environmental welfare point of view. We want to decrease the use of plastic in the food packaging industry, because in US, every household actually use lot of plastic packaged food, like lot of like plastic garbage every day, like 2 to 3 pounds of like every cycle, 2 to 3 days, 2 to 3 pounds of plastic they throw from the home. So it is getting a huge concern for the environmentalist. Yes, we need to reduce the plastic packaging for food to save this planet. So now, but my area of research is like, I am focusing on enhancement and encapsulation of nutrients efficiently in the food system. So in the second slide, I told you like we are eating micronutrients. So those micronutrients are like building blocks. I will not say building block. Those are micronutrients are catalysts for our metabolic activities. And those micronutrients are very essential for our health. So when we deliver them into food system, are these going into our system or going out of the system? So my specific area of research is food system functionality. So whatever food we are making, we are packing them and selling in the market that has actually good quality after like shelf life, before shelf life, or it is like just we are eating a calorie packet, just a certain amount of calorie we are getting. We are not getting all the nutrients we need for our body. So my research area of research, I did PhD in emulsion system. So what is the emulsion system and what are the food products we actually consume as an emulsion? So food system, emulsion system is a system consisting of a liquid dispersed with or without an emulsifier in an immiscible liquid, usually in droplets larger than colloidal size. So these are mixed and these are larger than in colloidal size, but oil and water is mixed. It may be wine and water, water in oil. So we'll come all this to discuss. So when I started my work in food science, so my professor was very interested to introduce, he wants me to introduce some of the Indian good nutrients, or I would say nutraceuticals, into the food system. So I studied a lot and then I decided, okay, people, our golden spice, which is turmeric. People of America, they don't like turmeric at all because they like the color, but the flavor they can't tolerate. So as you know, thousands of papers are available, like in the Google Scholar or in sci-fi, you will see, yes, this spice is really a golden spice. This is very good for our health. That's why Indian population is still now has less Alzheimer's and less other things like it's anti-inflammatory, antimicrobial. It has so many, like it's cardiac, it's good for cholesterol. It's like it decreases the platelet aggregation. So this is very good for health. So I have chosen golden spice for my research. So then I started working. Yes, if I want to introduce into emulsion system, then first problem was this is like immiscible in water. It is slightly soluble in oil. So now how can I introduce into emulsion if it is immiscible? So we first hypothesized we will make multi-layered emulsion system. So in that we will add one miscible one and then another layer on top of it, another layer on top of it, so that in this layer our curcumin is going to be trapped, and then this will give a system that will mask the bad odor of this. Like the people do not like, we like or this curcumin odor, so people will like it. So we started working with forming primary emulsion. So primary emulsion process making is not difficult. We just, and then secondary, tertiary emulsion, and we need to characterize this emulsion is good or not. So we did some particle size measurement, and then stability was observed. So this work I published in 2019. But the problem with that, I developed one system, and that is actually, I will not say that I made a really good multi-layer system. So our emulsion scientists, who are really like eminent scientists in emulsion field, McClemens and Decker, they have done a lot of experiments in multi-layered emulsion. So I tried their experiments in my laboratory, but really it failed. So if I am making a multi-layer system, then the particle size of the emulsion from primary to secondary, secondary to tertiary is going to increase. But here, when I took the data, I haven't got that. I haven't got any like increase of the particle size in my research. So, and it was also unable to carry my curcumin. So as you know, like when we just dissolve curcumin in oil and water, it precipitates. So if it is precipitating, that means my carrier protein and carrier stuff is not carrying my curcumin. So the project failed, but I published this in multi-layer system because antioxidant activity was high, and I used some other electrolytes like sodium chloride to stabilize this system. So what actually we wanted to do? So these are the 4 tubes I'm showing. If I make something like I want to introduce curcumin in this emulsion, second tube is going to my aim. I want to make the emulsion like second tube. So if I make a drink with protein, curcumin, and other flavors and sugars into it, then my emulsion should be like the second tube. People are going to buy it if they see this is like stabilized and this is like homogeneous. But these were not, because curcumin has a certain molecular weight. It cannot, like it precipitates, always precipitates. So now, then I have chosen some, okay, I want to make something good. So I have chosen 2-3 ingredients that I can deliver this into food system. So first of all, you know, in India, always healthy milk is very common. We drink healthy milk, which is very good for health. So, but healthy milk, people in US, they are not going to like it. So I started working with whey protein isolate. The reason I have taken whey protein isolate, because in food industry, always our aim is to make something from waste product. I will not say waste product, value added product. So as US milk industry, they make a lot of cheese, and all the like whey they throw. So these days, whey protein isolate, we are isolating from the whey protein whey, and this is for the people who drink like muscle building people like who are like athlete, they drink this whey protein isolate for muscle building stuff, but very costly. So I wanted to make something, yes, if I use this whey protein isolate and this curcumin, I want to make something good drink with this. That is going to be like superfood or like a medicinal food. I will say superfood. Okay, so that was my aim, to make emulsion like superfood for my work. Okay, so what was the rationale behind it? So as we all know, like curcumin is highly antioxidative. So one thing I would like to clear here, when we take haldi powder, that is not the pure curcumin. Pure curcumin is a little bit different because we are taking the active ingredient of the haldi. Just active ingredient curcumin we bought from Sigma-Aldrich, and then that purified product we are using for making my delivery system. So it has two forms, like keto form and enol form. So many chemistry people are here, they all know it. And they are at enol forms are at equilibrium, but becomes highly nucleophilic at pH above neutral and readily reacts with primary amines to form imine. So at this point, our hypothesis was, if we dissolve curcumin at a very little bit, like above 7 pH, and we try to just homogenize them or just mix them, and because of the nucleophilic reaction, this thing will actually allow to make some hydrogen bond formation with the enol form hydrogen bond formation, and it will carry my curcumin with it. So this is the structure I gave, like two forms, and during the electron-rich pi bonds is a source of nucleophilic site. So in that site, actually, this curcumin can, our hypothesis is that it can bind to it whey protein isolate. So now I'm giving a little bit a clear picture of it. So curcumin is hydrophobic and insoluble in the aqueous continuous phase. Insolubility limits its use. Also, like I used casein hydrolysate, which is a protein from milk and which is amphipathic, which is soap-like and more readily disperses curcumin agglomerates. So we have some previous data that show casein hydrolysate to be highly antioxidative in combination with WPI. So we have chosen this casein hydrolysate as a surfactant, natural surfactant. So we are continuing the same project. So then we use our main recipe, total recipe was like whey protein isolate plus twin 20, which is food grade emulsifier, casein hydrolysate, natural emulsifier, and we stabilized in water in oil nanovesicular emulsion. Systems with curcumin cargo. So that will be a cargo, like it can carry curcumin. So for preparing this, first we made coarse emulsion, and then we sonicate that emulsion to reduce the particle size a little bit. And then after doing that, we use high pressure homogenizer to make the primary emulsion. So when we homogenize the primary emulsion, actually we get the final nano emulsion. So nano emulsion is just when we break micro emulsion, micro small particles into smaller particles, nano size. Nano size is like from 100 to 500 nanometer size range, small particles inside the, I will not say called particle, these are droplet in the emulsion. So then last thing, after making, we do not stop here because we have to establish that my emulsion is superfood, my emulsion is the best so that I can sell it. So we need to do some like routine sciencey measurement, like yes, particle size, and then contact angle and zeta potential measurement. And we also measured antioxidant activity. So because particle size will confirm the nano emulsion, like the term we can use nano, yes, this is nano, and the contact angle and zeta potential actually shows your stability of your nano emulsion. especially Zeta Potential, and measurement of the antioxidant activity. Yes, we want to see when we are breaking all these small droplets into nano-sized, we are increasing the surface of each molecule, each droplet. So if in combination, you are increasing the surface area of reactivity of whey protein and curcumin. So, so that way, like we made sure, yes, now I have given plenty of space to the curcumin that you can react and bind with my whey protein. So in the next step, here I am showing a nano emulsion structure. So this concept actually we borrowed it from pharmaceutical people, because in pharma industry they are using nano emulsion to deliver their hydrophobic drugs into the system. So in their system, what they use, like here I am showing you the food system, but in their system, this droplet, this like oil containing hydrophobic rectangular small things are my nutraceutical, those are curcumin, but for them, this is the drug, the hydrophobic drug they deliver. And these are the surfactants, these are mycelis, it has tail and head. So it is like a surfactant, all these, and blue colored, like a sky colored thing inside is oil. So this is the structure we are making, this nano emulsion we are making. Now, for your knowledge, I'm again just explaining this. So we start from aqueous phase, and our oil phase is liquid, like polymer or organic solvents and drugs. And for my emulsion, what we used polymers of whey protein isolate, and organic solvents we haven't used anything. We used like buffer, and in place of drugs we used our curcumin. So then we sonicate them to get the secondary primary emulsion, coarse emulsion, a little bit small size one. And then this is the machine which can produce very high pressure. So I went up to 210 megapascal pressure. So huge amount of pressure it can create, and it can just break all these droplets into small size. So after breaking this machine is like nothing, just a pump. And when you just inject this solution, and they will go to a chamber, and that pressure is made, and all these particles will be broken, and you will have a like outlet. From there, you will collect your emulsion. So this is our nano emulsion. So after this state, nano emulsion, what we try to see if it is stabilized or not. So think about it, when you are giving high pressure to a molecule, and when you are giving up that pressure, what will happen to your molecule? So all these molecules immediately will try to coagulate. So they will immediately try to coagulate again. So to prevent that, we use that surfactant. All those surfactants are going to disperse all these nanoparticles and make a homogeneous mixture. So now, next step. So next step, we measure all them. So what we, to characterize nanoemulsion, so we measure particle size, zeta potential, hydrophobicity, because we are using some protein here, and measure contact angle, dispersibility, stability, and also if we want to establish that our curcumin is like quality of my curcumin is increasing, we need to do antioxidant activity. So these are the two machines I used. One is the Stansted, made by Stansted company, and these days so many like other industry grade high pressure homogenizers are available. Little bit costly. It was like $40,000 cost, and this one particle size analyzer. So I used that one too. So when the people came and selling this product to us, they are selling like you can measure anything, like dry ingredient, even we can use to measure this particle size analysis. Is a very good piece of instruments if you have. So you can do a lot of research under our food, even in chemistry. In this slide, I'm continuing like characterization of nanoemulsion. So we are measuring zeta potential. So I would like to tell our research scholars, so what actually zeta potential is. So zeta potential is when you are you have like salt and all stuff. Inside the emulsion, this negative and positive charge particles or positive charge like molecules are surrounded by your core of your droplet. So this is the like how much actually electric potential is there. The total electric potential is actually called zeta potential. So stability range of emulsion of zeta potential is plus minus 39. So if it is between like 39, your emulsion is going to be stabilized. So you don't have to worry, like, yes, you can keep this in shelf life for 1, 2, 3 or 4 months or 8 months, your emulsion is not going to separate. So, and the second thing is contact angle. We use in machine, we use this Young's equation. So when we drop off like emulsion on a surface, hard surface or solid surface, it makes a contact angle. So that contact angle also like measures, we measure that, and certain contact angle actually if your emulsion can make, that stabilize your emulsion. And also we measure solid to liquid interfacial free energy. So this is very important, because if the interfacial free energy is high, then your emulsion might not be stabilized. So this is the equation we use, like Young's equation. So here delta is theta is the contact angle, and gamma SL is the liquid solid liquid interfacial free energy, and gamma SV is the solid surface free energy, and gamma LV is the liquid surface free energy. So now again I'm giving here nano emulsion. So now our nano emulsion is ready. So yes, we have oil containing hydrophobic inside, hydrophobic curcumin inside. We have our surfactant and we have our oil. So now this is the graphical pictorial side, each droplet actually characterized in this picture. So now what actually we have got? We have these 6 types of, I have given 6 slide picture of electron, these are same scanning electron micrography of my emulsion. So we pictured all these emulsion, how it look like. So in this one, first one, this is with casein hydrolyzate and WPI. So second one is, this is in 140 megapascal, and second one B is in 210 megapascal. So you can see the difference in that when we apply 210 megapascal pressure, this emulsion, these droplets are already like, it's like smooth. And second one, this is the D1. This is also 140, but here we have given like a curcumin, casein hydrolyzate, and surfactant. So third one, so let me see this second one, what we have given. In the second one, we have given D, so C is with WPI 220, peanut oil, casein hydrolyzate, these are. And in the fourth one, we used same combination, only 210 megapascal pressure. And the last one, we introduced everything there, like 220 whey protein isolate, casein hydrolyzate, and 140 megapascal pressure. So it is giving a smooth texture. If we compare all these 3, the third one on the left hand corner is giving the best type of like texture under electron micrography. So also the result was the best in this one. So now we have a lot of data, like we have a lot of like data from this machine. So, and also like we did this experiment when we are doing it, all these like samples, we did it 0 day, like initially when we made this emulsion, second day, 4th day, 8th day, 12th day, 16th day. So gradually we did like 28th day. So because we wanted to see if the emulsion is stabilized or not, because what happened when we actually take data, and in the first day, even our zeta potential is showing plus minus 39. So we can conclude, yes, our emulsion is going to be stabilized, but in real life, it might not happen. The same, especially in food, when we work with food, just like molecular docking, We tried so many things in molecular docking, it's working, but in the bench top, it didn't work. So that's why we do like shelf life study or shelf life. So after 28 days, we characterized everything every day. So what we have seen, even after 28 days, our emulsions are stable. That was like, yes, breakthrough, yes, we have done it. So you see in those like curcumin, WPI, even all these are below 100 nanometer range. All these are 100 nanometer range and below 100 nanometer range after 21 days. And in the second one, which will be like stunning for us, it was because when we see the antioxidant activity, yes, antioxidant activity of these all things was highest in the 28 days. Even 28 days antioxidant activity was highest for all these. And if we compare them, so that was like 20, yes. Mean antioxidant activity relative life unit of the WPI, peanut butter and casein hydrolysate formulated nanovesicular vehicles and curcumin loaded vesicular vehicles were subjected to homogenization of 24 days. But I am telling you the result here. I think I haven't written here. It was like for Trolox, which was our control. Trolox is a standard antioxidant. It has RLU of 13.8. But when we did all this, our result was 1035 RLU. So you can see like from 13.8 to 1045, something like that. So it was like 1,000 times like more high antioxidant activity we can achieve if we make nanoemulsion and that is stabilized in a system. So that was, I published this work in a Journal of Food Science and Technology. So third project was to seek to same like emulsion system I used, and then it subjected to UV radiation. So now all you say, why you are doing UV radiation? So UV radiation is not good for health. So why you are treating your food in UV radiation? So the rationale of UV radiation was like curcumin in excited state intermolecular hydrogen atom transfer and curcumin react in ES-IS-HT. So ketoenol form of curcumin has been shown to be predominant tautomer, and curcumin undergoes E-sheet in various solvents when exposed to UV. This was our rationale, and we wanted to see, yes, this enol form and keto form, when it goes to E-shift, how the antioxidant activity actually differ. So after doing this was also a really good result. So after the first when we made it and we have exposed it in the first day, we do not see any significant difference in between these samples in the first after making. So and then we after keeping this 28 days, what we see, the samples we actually treated 15 minutes under UV radiation is giving us tremendous good result, and all three treatments from 0 to 140 megapascal and 210 megapascal are all significantly different, and they are giving best results for our emulsion. So that's what all in my like a normal like layman language I'm telling, so that all can understand. So that means if we make an emulsion and we nano emulsion and then we UV radiate it for 15 minute UV radiation, and we can keep this sample for a long time and antioxidant activity is high. So after finishing this project, I also published this one. So I saw after like my work, project like they made some products out of this nano emulsion and whey protein and the formulation I have given. They made some salad dressing, and they did some like we call it like human testing. Human testing is like sensory analysis. So after sensory analysis, what they saw, like whey protein can mask the bad odor of, like I will not say bad, I'm Indian, I eat a lot of like curcumin. So the pungent odor of curcumin. It can mask it. So people are liking it. So that means now in American salad dressing shelf, one product is there that is made with curcumin, whey protein, and all this stuff, and people can use it as a very superfood. So now for the for showing you how we can use different technology to make our emulsion or any food for the enhancement of our food product. One is microfluidization technique. Actually, these are old technique. Microfluidization technique was used I think 40 or 50 years we are using microfluidization technique for making nano emulsion. This is same kind. So we aqueous phase and oil phase, and we shear them in sonicator and then use the microfluidization technique, and then we get nano emulsion. Another technique we can use, phase inversion method. This was also old method. This oil phase and aqueous phase is like adding drop by drop and stirring in temperature, and then we use like conductivity measurement, and we can make nano emulsion out of it. So solvent dispersion method is another method we can use. We use the oil phase dropwise or polymer in a dropwise form, and then we make the emulsion and in a organic solvents. So for dissolving these immiscible drugs or nutraceuticals, what we are using an organic solvent, and we are evaporating the organic solvent using a distillator, and then finally we get the nano emulsion. Another is phase inversion composition method. This is also like aqueous phase is added to the oil phase under gentle stirring, and lipid and surfactant, and in case of drugs we use here our nutraceutical. Nano emulsion can be made. So the products we are and now after finishing all these, so I joined here and writing projects and getting grant. So now my main concern is like I am going to use all these nutraceuticals in the products like milk, butter, margarine, or fruit juice. So also like soup mix, cake batters, mayonnaise, ice creams, sauces, desserts, and then coffee, whitener. These products I can make with some nutraceutical so that American people can get some nutraceutical in their diet, especially thinking of COVID situation. So in COVID situation, one thing is very bad. One thing is like here in US, the people died because of obesity and diabetes and hypertension. So I am working with some protein and see their antioxidant activity. And I am, those proteins are plant proteins like indigenous, some sacha inch nuts. And I'm isolating those protein. I am isolating protein from rice. I'm isolating protein from different other small insects, so that these proteins actually have activity, like it has ACE inhibitory activity. So I'm measuring this ACE inhibited activity, so that if I get is something good, I will introduce it to the food system so that people can have this food as like medicine. So that like they are hypertension, if they are hypertensive, they can consume them and they can have good food. So as our addressing speakers were Dr. Register Sir was telling us, when we apply science as a scientist, what we should think, like how we can apply it for the betterment of human life. So my concern is that, yes, when I'm going to make something and think a bigger picture what I am looking for. So these days in US, there is a small corner in the grocery store. So there is a small corner there, you will see gluten-free food. So my concept is I'm telling my students, as New Orleans is a very vibrant city and very vibrant like for restaurants and stuff. So I told my students, why not restaurants for diabetic people? Why not restaurant for cardiovascular people, like cardiac health people? Why not some food for hypertensive people. So days are coming in the United States. So there will be corner like for each type of, that is called personalized medicine or personalized food. So people have specific problem and they will go to that aisle and collect their food and go back, and they will go back to home and eat them. So this is going to come. So as a food scientist, as we are like certified food scientists in the United States, we are trying to make all these happen. So that is our IFT goal. And also like as consumption of high protein, high animal protein is bad for health because of the problem of cholesterol, we are trying to make plant-made food more. So already like we are trying to make burger of plant food, like plant protein. And also I saw in Indian, some of the Indian companies like Go Food or something like that, a guy from Delhi, he's making all this meat product out of meat from plant product. So plant meat actually. So these are all we are trying to give the better life. And for immune booster, this is my area of research, and also like I'm trying to do a lot of work in immune booster thing. So here I just talk about curcumin because this was my PhD study. But all these ingredients, if you combine them, suppose curcumin, if I combine them with black pepper or nigella oil, I have seen this, the antioxidant activity becomes too high, and this works better in cell lines. So when we confirm all these things, like yes, in cell line, if we combine these two, this is going to enhance its immune, it will be like immunobooster, because nigella oil is very good for COVID. And also epigalactocatechin of the tea and the green tea, which is very good. So we are trying to make some of the system like I did with the curcumin and whey protein. We want to develop all these products, and we are working on these different projects at a time. We are giving our undergrad students each project so that we can characterize them and we can establish something. So now, what I'm telling our students, like there are plenty of opportunities, plenty of way of thinking. So what we do in your PhD is not everything. So when you become a scientist, you think all the needs of your field. Yes, in this field, I can do this. I can do this something better for my like career development. So just after PhD, so I never stopped in a single field. So I jumped from one project to another project. So my actually some of people, my buddy scientists, they call me adventurous. So yes, I'm an adventurous scientist. I like to jump from one field to another field, and I work really hard. So this was another, this is nutraceutical delivery system. This is the book chapter I wrote to know how we can deliver nutraceuticals in other system too. So this one, I'm showing you the difference between two systems. One is solid lipid nanoemulsion and nanostructure lipid carrier. So in this picture you see in the left hand side is solid lipid nanoemulsion, which is a nutraceutical delivery system where after storage, all these nutraceuticals are coming out of the system. But if we make nanostructured lipid carrier, in those after even storage, we can keep these nutraceuticals inside the system. So this was published in Elsevier book chapter. So another thing I'm following here also, like how to make microgel, how to make like nanogels in food system. So so for making this, we need to cross-link hydrophobic shell and hydrophilic and hydrophobic shell. So in the first picture you see, we are cross-linking, physical cross-linking of hydrophobic and hydrophobic shell and hydrophobic core and hydrophilic shell. This is forming like micelle. And in the second one, when we nanostructured them, nanogels, we are forming, nanogels, we are combining these two by cross-linking. So now, one thing we make sure here, when we make nano gel kind of thing, always OH group is there. So OH group has a very bad habit. It can make hydrogen bond, lot of hydrogen bond inside it. And hydrogen bonds are very fragile bond. So if you just take out of a little bit like temperature, if you heat a little bit of temperature, you give them, it will break, and it is going to be a watery thing. So your gel is not going to be the gel-like thing. So we are trying to make this also. So when you know your subject very well, so what happens? I want to tell you, students. So one of my friend was working with some gel for making jelly kind of thing. So he was adding OH, KOH, potassium hydroxide. So then he asked me, I'm using potassium hydroxide and I don't know why my gels are like, you know, after some time it's like making a liquidy thing. Then I told him, OH group is there. And then when you are adding too much OH group, then it's making hydrogen bond. So my topic here, like I want to tell your students or our audience, when we study by your heart and try to know what you are doing in each step. So characterize each step, then you can apply that in different field you work. So now, last adventure I had in Holy Cross. So I joined as an assistant professor. I'm an international. So it's really a challenge for me to survive. So actually from, as this is not a very big school, we actually received some fund from industry. So we asked industry people to give us some problem and we solved them. So this problem was given to me after my joining. So this was a coffee chaff problem. So in the coffee industry, they produce a lot of coffee chaff, which is a husky thing, husk of the coffee. And this chaff is a problem because it clogs the pipes, water pipes. It cannot be used for peat like garden soil or anything. It cannot be recycled. So this is a problem. So they said, if you can do something something with that. So I made a really good discovery with this. So what I did, this is the background. I'm not reading all this information step by step. So, or rather I will read it a little bit because this is a non-biodegradable plastic-based packaging material is increasing and growing environmental problem and safety issues have led to increased interest in the development of eco-friendly material. And coffee chaff is a waste product of the coffee industry. This is unique because it has lignocellulose more than 77.9%, which is insoluble. So my first concern is to make it soluble. So what I did, so objective was to make this a soluble product and then make kind of like paper, which is biodegradable. And I did it. So this is the coffee chaff. I used starch and glycerol also to make a film like paper kind of thing. So first thing was like I made a special kind of buffer. That is like I'm not disclosing the recipe of the buffer because it is kind of different. So that actually solubilized my coffee chaff. And then using that slurry with starch and starch and our glycerol, I made some kind of paper. So I'm showing you the results. I'm going fast. I think I have already covered. How many minutes I have, Devarati? Can you tell me how many minutes I have?
I'm not sure, but maybe we should find out.
I think 5 or 7 minutes I can finish my talk.
Yeah, please, ma'am, please.
Yes, definitely.
Yeah, so 5 and 7 minutes. So we did like thickness measurement. So this is like basic making paper making like characterization technique. So thickness of the paper I took, and I did like water solubility. This was the most interesting character I have seen. When I did this water solubility experiment with the paper I made, I saw like after I treated this in 25 degrees Celsius for 25 hours, but it is zero, which doesn't have any starch. It was stable and it was strong. It didn't dissolve, and it was like semi-plastic material. So now I am using this semi-plastic material. So I tested on water solubility count. I tested like vapor permeability count, everything. It was color measurement. I did all the research here, and moisture content. So best thing what I have got, yes, this is a semi-plastic material that can be, and biodegradable, that can be used to make food wrap kind of thing. So that like, yes, this is not a plastic, because for food wrap, we use plenty of plastics for making food wrap. Also, another product I'm making with this, I am growing some mushroom on it, and it's growing very good. This is going to, I'm sure this is going to give a good result. So another, a few things I would like to share. So when I do this COVID research, also the next plan of work is like the emulsions, whatever I'm making, and all these proteins, I want to show, I want to see the digestive pattern of nano emulsion. That work needs to be done, and it can be done using like a gastric juice like simulation study, or we can make some like mechanized like a human artificial human digestive system and see the bioavailability, because whatever we eat, bioavailability is the main thing, like how much bioavailable that particular micronutrient is in your body, and absorption pattern is the main thing. So I want to study that, and all these antioxidant thing like from all these plant polyphenols and flavonoids are my like interesting field of study, because you will see in the Starbucks they are selling a health drink which is like $5 cost, only with some hibiscus flower and some blueberries. So they are making money just drying some flowers and some blueberries and making a drink. So these things can be a very good like industry approach, like even for India. That's what I'm thinking, like if we can actually teach our students all these making product kind of thing, they don't have to look for job, they can make their own starter company, like entrepreneurship. So this kind of many like idea I'm right now doing, working with some like non-profit organization to teach our community. Also the reason I'm staying here, there are many immigrants here, and also after the like I think Bush has this immigration pact and stuff, 50,000 immigrants are here. So we are thinking, we are already like writing a grant for NSF, because we want to actually educate them by teaching these technologies and teaching this stuff, so that they can make some money to survive in this unknown world for them, because United States is new for them. So those are the different like community development program we are doing under Holy Cross, because this university is a small university, and these are like governed by Mariantis of Holy Sisters. They started this university in 1960s time, and they did really good work. And this is not the just university just for teaching and stuff. This university actually does a lot of community development work for the poor people. So I'm happy that I'm working in a Holy Cross University, because I'm also like this kind of person. So my another plan, last slide, I think, Devarati, this is the, so two things I'm focusing on. One is like new product development with plant-based food ingredients, and second is plant protein. So I am trying to isolate protein, and my food research lab is made with all these protein characterization instruments I'm buying, Keldal and different kind of like micro analysis stuff. So this is all, this is my references. So in conclusion, I would like to say, our students and all audience, so when we do research, anything like how I just see what benefit I can make out of it, how I can sell my concept, and the solid science behind it. This is biotechnology. Biotechnology, the definition of biotechnology is we are using bio things, like natural things, for the development of human being and the population. So this biotechnological approach, my approach is like food and stuff, but any approach like Devarati is doing a good research, And other people, many other people are doing all this sequencing stuff, like changing stuff, all stuff. Like these are really interesting, and this will bring us in a field that we can live in a better world, and we can face this COVID crisis. We are really good fighter, so we will combat all these things. Thanks. Now question.
Thank you, Dr. Suma Mukherjee. So before starting question hour, I'll just request my friend, Dr. Anjaneyadu, to give a certificate of appreciation to Dr. Suma Mukherjee. So please accept it, ma'am.
Oh, thank you. Thank you very much.
Yeah.
So how can I accept it? Do I need to? Okay. This is, yeah, thank you. Thank you. Just send me an email so that I can. Yeah. Thank you.
Yeah. Sure, ma'am.
Thank you very much. I appreciate it.
Yeah. So really, ma'am, you are an adventurous scientist.
Yes.
As you said, you started your talk with your very impressive, motivated, about your career, especially for those who plan for research career, want to think out of the box to develop different materials with the help of biotechnology and natural food sources. You started with major and minor nutrients. What is the importance of diet in western side and eastern side, and how it is cooked actually? And in that pattern, how nutrients are getting repeated in, especially in our Indian context. That is really good. And even you have thrown light on global scenario of food market, where it's really amazing to know that every minute a new product is introduced. It's really good. And need for decrease in use of plastics, really that is environment concern, and we really all of us have to concentrate on that one. And one important thing regarding your research, that is a way all these days what we are thinking that it's a waste product, but it is not a waste product. It is a value added product. That is really good, ma'am. And whey protein, how to isolate and how to make it as nano emulsion particles. And with regard to your what do you call, ma'am, research, you said you are a little bit struggling about how to stabilize the beads actually, and I don't know if I'm correct or not. Normally, already established techniques like using calcium chloride or calcium based can stabilize the beads, and I think because adding calcium to the food product also, again it will be added nutrient advantage.
Yes. Yes.
So yeah, so that might be helpful if I'm not wrong.
No, you were absolutely right. Thank you. Thank you very much for your advice. I will keep it in mind and I'll apply this in my research.
Thank you, ma'am. And now I request our research scholar, A. Chetana, to take up the questions actually which are posted in the chat box. And even if any other questions are there, they are welcome to ask Dr. Suma Mukherjee. Over to Chetana, please. Hello.
Hello.
Yes. Yes.
Am I audible to everyone?
Yeah.
Yes, you are.
Good afternoon, all the dignitaries present over here. It's a very informative and interesting, ma'am. Personally, I have two questions. Ma'am, in a nutraceutical delivery system, whether all the active groups of proteins will retain their functionality? Means how we can check the active groups, whether they are active or they become inactivated, or what happens to them?
So actually the question you are asking about the protein or about the nutrient?
Nutrient, ma'am. In the nutrient, if protein is there or enzyme is there, whether the active groups are retaining their activity.
So in food system itself is a dirty system, we call them. So when we combine with protein, lipid and carbohydrate and all stuff, it is difficult to see the active part of it. So that's what I mentioned in my talk, that if we try to understand it, we use the molecular docking technique, ma'am. And then actually, if we do that and we understand, yes, this part is going to join with this part, or this part is going to not join with this part. But what happened in the benchtop, we do not get this kind of result. Honestly, like when we do it in the, we cannot see it, because that's why we call this like system as a dirty system. Sometimes it works, sometimes it do not. But the measurement we are taking, like antioxidant activity, acid inhibitory activity, or like functionality and stuff, bioavailability or dispersibility, for in terms of food characterization, we can do everything. But I talked to my professor about it, like this, how molecular docking we can see. So she told me, she's an eminent scientist, she told me, no, Soma, we can't do it. In food system, we cannot see that. But for like chemistry kind of work, if we do like in chemistry lab, yes, you are working with just curcumin and you are working with just one protein, you can see using molecular docking. Same thing also did with like for COVID situation in like Nigella's work, and different kind of like phytochemicals. They have seen all these like with COVID protein, COVID like that particular protein COVID has. And so they actually sliced all this protein out from the virus, and then incorporated into a E. coli, and then they replicated it and isolated that protein, and that protein they used to treat all these chemicals, all these like bioactive chemicals. So that's why they see like epigalactocatechin and Nigella are very good for COVID. So I'm really not sure about the question you asked. So that's this much answer I can only give you. Thank you.
Thank you. Ma'am, one more question from my side. That is whether we can use the same immobilization method, ma'am, immobilization process for the entrapment.
Yes, we can use same method.
Okay.
Yeah, we can use the same method. No problem. Just we just shift the drug to nutrient. We can use the same method.
Okay.
And always like last part, we characterize them. How is stability is there, or like any adverse toxicity is there. We use HPLC and stuff to see if any other ingredient is like producing that is toxic for health. So we do all stuff before like telling, yes, this is working.
Okay. Thank you so much, ma'am, for your answers. And it's my pleasure to take part in this international webinar. Now the session is open for questioning. All the audience, you can post your questions in the chat box. And already chat box have one question. Hello, ma'am, very informative talk. Recently, scientists have proved the traces of plastics in human blood, which has adverse effects on health. What may be the reason and how can we prevent that, ma'am?
Yeah, actually, yeah, this is like a major concern is going on. University of Minnesota, they have seen also in the bottled water we are drinking nanoparticle plastics there. But I saw one presentation, I think recently I saw it. I cannot exactly tell you the paper, but they have seen like now we can isolate plastic particles using a chemical that actually coagulate all the small nanoparticles, plastic particles from the drinking water. So this is new technology, new method is coming. I'm very like positive about that new in the coming few years, we're going to get this kind of like chemical that we are going to use to get rid of all these nano plastics from the bottle. But plastic bottles are not the only problem here. Problem is with the environment. Nanoplastic is everywhere. So till now, as far as my knowledge concerned, I have seen people are telling, yes, this is not going to harm us. And especially the Indonesian APO group, they have done a lot of work on this like plastic pollution and this stuff on animal study. I seen, yes, there are some adverse effect there. But till now, I think government and all like bureaucrat people, they are not making us panicky about this, because we have to live this in the world. We cannot go to the other planet to live, and plastic is everywhere. So best thing, they are trying to reduce the use of plastic, recycling process and plastic economy, and these people. I think some good outputs are coming here. Also, I saw like 50 people, 50 scientists in the US, all over the world. They made a forum. Actually, they are trying to degrade plastic somehow. So this is a huge work is going on where I think more than 200 people actually joined in this group. They are working to biodegrade this plastic, degrade this plastic. So some new insights will come out. Thank you.
Any other questions?
Yeah, ma'am. I think there is another question. Was there any particular species of? Hello. Yeah. There is another question, ma. Was any particular species of turmeric chosen? Did you?
No. Here I haven't done with the species of turmeric. So only curcumin, actually the purified active component of turmeric was chosen, and we bought it from Sigma-Aldrich. So to see the hydrophobicity, that can be actually we can deliver this the pure active component as a pure active component. But itself turmeric powder, if we want to like introduce in milk, the way we drink a healthy milk is fine. But even if you just do this experiment at home, just put 1 teaspoon of haldi in your milk, it is not dissolved totally. So you will see some precipitates are there. So that to get rid of that precipitate, we do not like any product that has precipitation. So that's why we did this research, like yes, pure curcumin, to see pure curcumin only. So no species we haven't done that. Because all species has, another research I did, I haven't published it yet, we just submitted that paper. We actually, we have grown some turmeric in US soil. And then we have seen that curcumin content is very low. When we are analyzing them in HPLC, curcumin content is low as compared to Indian sample, what we get in the market. So these are these things we are trying to introduce in our like agriculture system, that yes, we can produce this turmeric and introduce in our food. Lots of work are done, but I have seen turmeric content, like curcumin content is very low in those species like we have grown here.
Okay, ma'am. I know it's all over for you, ma'am, because for us it's a day, but for you, we know it's past 12, I think. So we are very sorry to disturb you at this time. So last question, ma'am. Like you said about coffee chaff, which is a a waste product from coffee industry. Can we use that coffee chaff to layer, what do you call, paper glasses? Because normally what they do is they layer thin fine plastic actually, which is also very.
That's what we're trying to make actually. That biofilm I made actually, this is semi-plastic. That's what we're trying to make. Like we're trying to make all these like trays, small trays, which has like foam, but it can make a layer. And then we can put a top of it small plastic, that we reduce the use of plastic. That is our main concern. So we are doing it.
Because so when you put hot drink actually in that one, hot water or milk and all,
yeah,
you get that coffee flavor where you can enjoy it.
Yes.
Yeah. So thanks a lot, ma'am. Really, so wonderful actually. Very good, very informative talk. And our another speaker also is waiting. I request
Thank you very much. Yeah, I will be here whole. I'm going to listen to other speakers too.
Oh, great. Thank you, ma'am. Thank you. Thanks a lot. Yeah. So now I request my colleague and friend, Dr. Kishore, to introduce the second speaker today. Thank you.
I'm very much delighted to introduce our second speaker, Dr. Shizwe Low. Dr. Shizwe Low is a senior lecturer in Biotechnology and Food Science Department at University of Johannesburg, South Africa. He holds a BSc and BSc honors degree and a MSc in biochemistry at University of KwaZulu-Natal. He then moved to Stellenbosch University, where he completed his PhD in 2017. He has worked at RCL Foods as a management trainee in the consumer division in 2016. In 2017, he joined the University of Western Cape Institute of Microbial Biotechnology and Metagenomics as a postdoctoral researcher. He then joined the University of KwaZulu-Natal as a lecturer in August 2018. Currently, he is a senior lecturer at University of Johannesburg. Dr. Sizwe's initial research interest was in biofuels and has since changed his focus to secondary metabolites for endophytic fungi, looking at the potential compounds with antiviral, antibacterial, and antifungal activities. He published 7 research peer-reviewed articles. Dr. Sizwe's research is funded by National Research Foundation and Poliomyelitis Research Foundation. I now invite our second esteemed speaker, Dr. Sizwe, to enlighten us with his talk. Over to Dr. Sizwe.
Good morning, good afternoon, those who are in the afternoon.
Yeah, very good afternoon, sir.
Can you see my screen?
Yeah, yes.
Okay.
Put it on screen. Presentation mode.
Full screen, sir.
Full screen mode.
Yeah, I'll do that.
Yeah.
Is it fine now?
Yes, please.
Okay. So greetings to you all. Let me switch off my video. So greetings to you all. As I've been introduced, I'm Dr. Sizwe Ndlovu, and today I'll be sharing with you what I've titled Finding New Anti-HIV Drugs from Silent Fungal Biosynthetic Gene Clusters. So thank you, Prof. Deparati, for the invite. I appreciate it. So I'm from South Africa. So it's in the south of the African continent. So in South Africa, we are about 60.7 million people in population. That is the last year's stats. Probably we're reaching about 65 now. And then in Johannesburg, we have 6 million people. And then of that 6 million, 0.8% is contributed by the University of Johannesburg student. So I'm based at the University of Johannesburg, the Doornfontein campus. So in the Department of Biotechnology and Food Technology. So as the speaker have introduced me, so I started in yeast genetics and biotechnology, and then I moved to the biofuels where I was looking at cellulase enzymes and the process engineering. Then I went on to a short stint in the industry where I was a management trainee. And then from the industry stint, I went back to academia where I did a postdoc. And then it is in this postdoc that I started to fall in love with these natural products. And when I joined the University of KwaZulu-Natal, I started a research program on fungal natural products, which I'm still continuing with. So we have called ourselves the Fungal Genomic Research Group, me, my student, and the collaborators that I'm working with. So the projects that I'm currently working with, it focuses on the on the opportunities and threats presented by the fungal kingdom. So in the first initiative, we're looking at the fungal antimicrobial resistance, focusing mainly on Candida auris, but we're also looking at other environmental fungi, and then looking at their resistance together with other collaborators. Then the second project, we interested in drug protein interactions. So we identify drugs, and then we identify protein targets, and then we do in silico studies. And also once we have found like a good binding patterns, then we move to in vitro studies. And then what I'm going to talk about today is my interest in fungal natural products or fungal secondary metabolites. So these are fungi that are associated with medicinal plants, and we are looking at antibacterial, antifungal, and antiviral activities. But for today, I will talk more about the antiviral activity project that we have. So what are natural products? So why natural products? So the increasing bacterial and fungal and viral resistance and the lack of new discoveries is a cause for concern for the public health. So unfortunately, this dramatic rise in resistance this dramatic rise in pathogen resistance and emergency of new pathogens cannot be counteracted by the molecules that are currently in the clinical pipeline. So there is a need for new intervention or finding new drugs from different sources. So one of the sources that we know in history that it's a good source for active metabolites are natural products. So a natural product which possess an enormous structural and chemical diversity that cannot be matched by any synthetic libraries of small molecules. So these continue to inspire novel discoveries in chemistry, in biology, in medicine, because when we look at the clinical drugs that we have, more than 60% are either natural products or derivatives of natural products. So the actual work on natural products, or the interest on natural products, it started by the work by Alexander Fleming in the 1920s, where he discovered penicillin. Then penicillin was subsequently produced at large scale by Florent and Chain in the 1940s, which laid the foundation of natural products. Then we see here in the period of between 1950s and 1960s, which was termed the golden age of natural products. There was a lot of discoveries mostly from soil and mainly the bacteria, the microorganism was actinomycete at that stage. So they discovered antibacterial, anti-cancer, antifungal, antiparasitic, and immunosuppressant agents, and all other kind of drugs that you can think of. But then this was followed by a decline in natural product discoveries, and there are so many factors that contributed to this decline. These include the rediscovery of known compounds and technical challenges, which included the purification and structural elucidation, and failure of combinatorial chemistry to deliver promising new drugs. So we saw that in those years, or even now, the pharmaceutical companies, the big pharmaceutical companies, they withdrew their interest in natural products, and they moved on to synthetic chemistry, which have promised to deliver more exciting drugs, but that these have not even met the challenge. So interestingly, in the 1990s, we witnessed a ramping up. So there was a ramping up in large-scale microbial genome sequencing projects. So these have dramatically improved our understanding of the genetic and enzymology of natural products by biosynthesis. So the whole genome sequencing has shown that molecules that have been discovered in the initial screening initiatives, they only represent a tip of the iceberg. There are many genes that, many biosynthetic gene clusters that we have not explored. So this is particularly true for the endophytic fungi, which are microorganisms that colonize the intra and the intercellular regions of your healthy plants. So these fungal species, they live in a largely mutual relationship with your plants, where they produce, where both the species, the plant and the microbial symbionts produce different compounds in response to environmental challenges, such as your invading pathogens, your nutrients, and other factors that can present in the environment. So it is believed that these symbionts have co-evolved with the plants to share the biosynthetic potential, such that the endophytes could produce similar compounds to their host plants, which is something that we can really look at in terms of preserving the plants, because if we can find similar products from the endophytes, that means that we don't have to over harvest the plants, because and then the endophyte is a good alternative because it's sustainable. You can manipulate it. You can manipulate the fungi in the lab cultures, and then you can also do heterologous expression and all sorts of things with the fungi. So some of the compounds that were first found in the plant include your first one, which was Taxol, which is an anti-cancer drug, which was later found to be produced by the endosymbiont of the fungal endosymbiont of the plant. And after that discovery, there were many other compounds. I can name a few, but I've just listed two, which are antiviral compound, alteratoxin, and then malperoxide C, which are antiviral compounds that were first identified from a fungal endosymbiont, from a plant, and then later from a fungal symbiont, endosymbiont. So this secondary metabolite released by endophytes as host defense against antagonists and can be exploited for therapeutic purposes. So this provides a good selection criteria for the plant, because so in our environment, in Africa and also in Asia, because of the rich history of traditional medicine, we have a starting point of selection of our plants. We can select our plant based on the traditional medicine history that we have, as in order to have high chances of isolating endophytes with which produce novel compounds against the disease. Okay, so when we started this project, we started with antibacterial assays, looking at antibacterial, but I'm not going to talk about that today. What I want to draw your attention to is the antiviral compounds. So we, my initial collaborator was Dr. Nompumelelo Mkhwanazi from the HIV pathogenesis program at the University of KwaZulu-Natal. So we started this initiative together. So she is a virologist, so she has been part and parcel of developing the viral assays that we use. And then we got a number of students, but I want to present on the interesting finding that have been discovered by Mr. John McIntyre, who is our master's student, who he's busy with the write-up at the moment. So why HIV in South Africa? We have about 7.7 million people living with HIV, and then we are a country with a pandemic of HIV, so we have the largest antiretroviral program in South Africa. So we are making inroads with the HIV because we are about to meet the 90-90-90 targets, which have been set out by the World Health Organization. So at this point where we are sitting, about 92% of people in South Africa are aware of their HIV status, thanks to all these programs that have been rolled out to destigmatize HIV, to encourage people to get tested and to know their status. And then 75% of this 92 are on HIV treatment. So, and 92% of which are virally suppressed. So if you take your treatment appropriately, your viral load will be repressed, and there are very less chances that you can transmit it or transfer it to your partner or to another person through maybe a sexual intercourse, because your viral load is repressed. So this is a good development in South Africa that we see in South Africa in terms of, as far as HIV and AIDS is concerned. So let me draw your attention to the HIV life cycle. So first, the viral particle or the virus attaches to the cellular membrane. So this will be our cellular membrane for today. So it attaches through a viral envelope, which is a protein called, a glycoprotein called the GP120. And then this GP120 binds to the extracellular domain in the cellular envelope, which is your CD4 plus receptor. So the binding of the CD4 with the GP120 causes the conformational change in the CD4, in the GP120, which exposes another binding site for a co-receptor, which we know it as it allows the presentation of the co-receptor binding site. So this exposes the GP120 to like the chemokine receptors, which can be CCR5 and CXCR4. So the binding is mediated. So the binding to these receptors is mediated by a Vitry loop sequence and a glycosylation. So this causes further configuration changes which take place and expose the domain of the second protein, GP41, and then once the GP41 binds, it brings the virus much closer to the cell membrane, resulting in the fusion of the virus as it enters the cell membrane. So in the cytoplasm, when the virus is in the cytoplasm, it releases the viral RNA. So the viral RNA then is reverse transcribed to viral DNA. So once the viral DNA is reverse transcribed, the viral RNA is reverse transcribed to viral DNA. It is subsequently integrated into the genome, the host genome, by an enzyme called integrase. So this leads to chronic or latent infection, as we know that the HIV virus, it hides in your genome, or it integrates into your genome, and then it becomes difficult to targeting using the currently available drugs. So some of this proviral DNA that is integrated into your genome is able to be translated to mRNA and then to polyproteins. Then we have other sets of enzymes, which we call them proteases, which cleave these proteins into small proteins, which then are infectious, and then they can go on to other cells and cause a viral progression. So currently we have a regimen of drugs which include your reverse transcriptase inhibitors, your integrase inhibitors. We also have your protease inhibitors and your fusion inhibitors. So the current drugs include all these components in one treatment or in different treatments. So you take your drugs which include the cocktail of all these inhibitors. But then what we have seen over the years is that these drugs are losing efficacy because of a number of challenges. So one of the challenges that people who take this medication, they do not stick to this medication, they default, which lead to increasing resistance, and then they will be allocated to a second regimen, which is the last option at the moment. And then when they default in the second regimen, then it becomes difficult because there were no therapeutic options for them. And then the other problem that we currently have with HIV is that the latent provirus that is integrated into your genome, there is no current medication or drug that targets that proviral DNA. So it remains latent in your cells, and then it can be activated at any point. So that is why we have been unable to eradicate the virus at the moment. So we've taken an initiative to look at the fungi, which is known to produce, which have been known for over years, for all these years, to produce several antimicrobial compounds. At the moment, there are no antiviral compounds that are produced by fungi in the clinical pipeline. Although this is largely contributed by many factors, which include the lack of research because of maybe lack of access to facilities to carry out the antiviral assays, and also the antiviral assays are not well developed as antibacterial assays. So that is why we have not seen many initiatives looking at anti-HIV or antiviral compounds. But we have recently done a review where we look at anti-HIV compounds produced by fungi, and we found a subset of these, and we've realized that fungi also hold a promise as a source of antiviral compounds. So that's when we started developing these assays for screening the antifungal agents from the secondary metabolite, the fungal secondary metabolites. So what are these secondary metabolites? So these are encoded in genes that are arranged in clusters. We call them biosynthetic gene clusters. So they can be classified into polyketides. So some of the polyketides that you may be aware of is your cholesterol lowering drugs, which is your lovastatin, and your toxins, like your aflatoxin B. We also have a group called non-ribosomal peptides. The most prominent one is penicillin. We also have hybrid groups, which is a fusion of your polyketide and non-ribosomal peptide. And one of these is a newest antifungal drugs. It's echinocandins, which falls under this class. And then we have terpenoids. One of these classes is your gibberellic acid. Okay, so the most often, the problem is that most often these biosynthetic gene clusters, they do not produce appreciable concentrations of metabolite during cultivation in standard laboratory conditions. And in some cases, they are totally silent, or they are totally not expressed, and we call them silent or cryptic genes. So we currently do not know the exact environmental triggers that lead to their expression in the natural environment. We also do not really know the regulatory systems that govern their expression. The information that we have currently is poorly elucidated. So it's difficult now to control the expression of this, because we don't know what are the interactions in the environment that lead to the expression of these compounds. So in the recent years, there are various methods that have been developed to activate these silent biosynthetic pathways. So these include your heterologous expression. So once you've identified through a chemically guided assay, you identify the genes that are responsible for a specific phenotype. Then you will clone that gene, which usually is a stretch is a long stretch of genes which are clustered together up to about 100 kilobases. But there are methods that have been used in literature to clone these big clusters. So you do heterologous expression, you take that gene, you move it to a heterologous host that you can control, that you do have molecular tools for it, and then you are able to study the phenotype that is produced. So the other system that have been developed is systematic alteration of cultivation media or cultivation parameters, which we call them one strain, many compound approach. So in this approach, you introduce various conditions, growth conditions, to mimic what is happening in the environment, and hoping that in one of the cultures you'll find an altered expression of your secondary metabolites. So the other technique that have been used is co-culture or co-cultivation. So in this case, you co-cultivate your microbe of interest with another microorganism. So again, you're mimicking what is happening in the environment, because when these are in competition for food, space, and survival, then they will start attacking each other, producing these secondary metabolites, and then you can harvest these secondary metabolites. So there's another one, technique, which we call it ribosome engineering. So in ribosome engineering, you make changes or mutations in the components of the ribosome or the RNA polymerase that results in increased production of secondary metabolites. Recently, there have been a high throughput technique, which we will call it HITES. So the HITES, it's an abbreviation for high throughput elicitor screens. So in this approach, which was developed by Xiu et al. In 2017, so a reporter gene is inserted inside the biosynthetic gene cluster of interest, and the resulting strains is screened against a library of known chemical elicitors in a high throughput manner to find the candidate elicitor. So what are these elicitors? Can be your known antibiotics that have been used as part of the elicitor library. So you're using small molecules to find other small molecules, which is quite interesting. So the libraries, they have been shown to serve as modulators of silent gene clusters at sub-inhibitory concentrations. So before you use these modulators, these elicitors, you have to establish the MIC first, their MIC first, and then you have to use sublethal concentrations. So lastly, I would like to draw your attention to the chromatin remodeling. So this is one of the techniques again, but what is important, what is good about this technique, you don't need to know the biosynthetic gene cluster of the organism. So you can use it for environmental, for screening your environmental isolates without knowing any genetic information about the isolates. So in this technique, the fungal cultures are supplemented with histone deacetylases or DNA metylase inhibitors. So this mimic the epigenetic alteration of genotype in the environment. So let me unpack this further. So we recently did a review on the elicitor, on the epigenetic elicitors, which we published this year with one of my PhD students. So this concept of chromatin remodeling is basically based on the knowledge that fungi, the fungal biosynthetic gene clusters, are organized mostly or grouped in locations that tend to be near telomeres. So if you, for people who understand the fungal genomics, so if the genes are located near the telomeres, so they will be transcriptionally silenced due to their location. So this results in histone acetylation and DNA methylation having a large impact on their transcription of these loci. So the histone acetylation and DNA methylation are some of the examples of your post-translational modification of genes that have been elucidated. But there are other post-translational modification that occurs in the chromatin, and then most of them we don't really know what is their function at the moment. But HDEX and then DNA methylation, we know a bit more about them. So what happens is that your chromatin is a highly compacted structure, and then in this high compaction, it is post-translationally modified by different systems, which lead to a heterochromatin landscape. So in this heterochromatin, the biosynthetic gene clusters are not exposed, so are not transcriptionally accessible, so they are transcriptionally silent. But then if you use HDEC inhibition, or you delete the HDEX, then you open the chromatin, which we call now a euchromatin with altered gene expression, then you can access the biosynthetic gene clusters. So this technique is based on this opening the chromatin. So the first initiatives, what they did was to knock out the HDEX. So they will knock out the HDEX and then lead to euchromatin, and then this will lead to an altered secondary metabolite production. But after that, those interventions, they realize that you can use small compounds that are HDAC inhibitors without knocking out the HDACs. So these compounds are your valproic acid, your sodium butyrate, your trichostatin, and many others, which perform the same function as knocking out the HDAC. So it represses the HDAC and then lead to an altered gene expression. So while we were doing the review, we established that not all HDAC inhibition lead to production of more secondary metabolites. In some cases, it can lead to repression of secondary metabolites, where we see that the untreated fungal culture will have more secondary metabolite profile than the treated fungal culture. So it's not always an overproduction. It does not always lead to overproduction. Recently, we have seen the advent of CRISPR-Cas-based genetic engineering strategies, which have provided new possibilities for precise genome editing in filamentous fungi. So we've got quite a few now which have been adapted to filamentous fungi, thanks to all the researchers who have been contributing all these tools. Initially, the CRISPR-Cas was not designed for filamentous fungi, but it has been adapted as molecular tools for fungal genome editing. So the most interesting for today that I would like you to look at, the first one is your gene silencing. So the gene silencing. So in this technique, the CRISPR technologies, in this CRISPR technology, you mutate the endonuclease. So the endonuclease is the Cas9 endonuclease. So once it's mutated in one of its domain, then it loses its catalytic activity, but not precise pointing of your nucleotide for whatever genome editing specification that you want to do. So the deactivated Cas9 is then used to target the transcriptional initiation or elongation regions. So this reduces the amount of mRNA that are generated through competitive inhibition, so leading to gene silencing. So the second one is gene overexpression. In this one, in this gene overexpression, the CRISPR-based transcriptional activation of a target gene, the domain or the entire protein is fused with a deactivated Cas9, which facilitates the recruitment and assembly of the RNA transcriptional machinery to the target site specified by the single guide RNA, and thus driving an increase in gene expression. So these are the tools that you can use for in fungi as well. Again, these ones you need to know a bit about you need to have a good sequence in order to perform the CRISPR-based intervention, but it provides a good platform for overexpressing or silencing genes to perform partial or full functional genomics in filamentous fungi. So what we've done over the last 3 years, we've collected some plants and we've isolated some fungal endophytes, and then currently at this point we had 49, but currently we have more. I have not had time to sit down and calculate how many, and also do an account of what we have as well. But this was the first collection that we did, and then we've managed to identify a few with my few students that I've worked with, and the strategy that we mainly use is the basic strategy where we isolate the endophytic fungi. We first do a preliminary screen because we usually get more endophytic fungi, and then we don't have the budget and the manpower to do the identification for all of the fungi that we have. So we do a screen. It depends on the nature of the project. If it's an antibacterial project and then we find a good hits, then we send those promising endophytic fungi for sequencing if they have not been sequenced before. But it depends on the screen that we do, antiviral, antibacterial, or antifungal. Then we do identification using molecular tools like your internal transcribed spacer sequence, which is I think it's the golden standard for identifying fungi. And then we do activation. So normally our activation have been based on epigenetic modification, but with advancing tools, since we now have the advanced tool, we are moving to CRISPR-Cas based activation. And then after activation, we do crude extract preparation. So now we want to trace the metabolites, the fungal metabolites, and then we again do a preliminary screening activation assays. So when we find hits again, you can see that in every step we stop and do an activity assay to check if we still have, we still retain the activity. So if we find a hit in our preliminary screens, then we move to large scale fermentations, where now we produce in bulk. Remember that when you move your fermentation from a small scale to a large scale, the parameters might be the same, but the compound might be different. So there's a lot of optimization from moving from a small scale to a large scale. So you optimize and do several assays. So it's a lot of going forth and back, and until you find the good conditions that will lead to the production of your desired secondary metabolites. So this large scale is followed by, once you've confirmed the activity, you follow the large scale fermentation by your solid phase extraction. Basically, this is to simplify the metabolite profile. As you know, that fungi produce a number of metabolites. So with the solid phase extraction, you simplify the metabolites again. In every step of the extraction, you test for activity. And then once you have simplified fractions, you then submit your fractions for identification, for chemical identification. This particular moment we have used GCMS, but we are more open to use other techniques. So that basically the scheme of things that we follow. So one of the assays that we have worked with, though there are so many assays now that we have developed in terms as far as antiviral activities concerned. So the first one is Luciferase-based assays. So this uses TZMBL cell lines. So these are basically your HEC293 cell lines. They express CD4 cells, the CD4 cell, and the two core receptors, which is chemokine receptors, which is your CCR5 and your CXCR4. So they also express the long terminal repeat, the HIV long terminal repeat, which is your promoter sequence. It also expresses the BCAL and Luciferase, which give us the output if there's viral inhibition, or which we can use to measure viral inhibition or viral expression. So the initial screens that we do, we first infect with the pseudovirus. So we first do our screens with pseudovirus. So this is the virus that we construct in the lab using certain components of the virus. So we design a pseudovirus. So we use it as an initial screen. Then when we have found a promising hit, then we move to patient samples. So cohorts which have donated their samples, where we have different strains of HIV or different HIV with different resistancy levels. So we test also on that, using that. So this is one of the assays, but we have developed about 4 assays now that we work with. So we are aiming to develop more, because we want to, this one, it's also only look at the replication, the viral replication, but we also want to look at the protease inhibition, the integration inhibition. We also want to look at immunomodulation and all sorts of things that we are interested in with the positive hits that we have found. So unfortunately, I cannot share the graphs from a student. I did have a talk with him this morning. He said no, because we have not published the results. We're in the process of publishing those results. But this is the summary of what he did on his screening. So he screened a number of endophytes. Obviously, we limited it to about 10 endophytes because the antiviral assays, they are expensive. Each kit will cost you a lot of money. I don't know in Indian currency what is it, but in South Africa, each kit will cost you about 100,000 per kit. So, and then you have to kind of like limit what you screen. So we screened about 10, and then we decided on the second screen, our initial screen, then on the second screen, we reduced this to a few, I think about 3. And then what we found is that, so we had treated fungal endophytes and the untreated. So the treatment that we had were 2. We started with valproic acid and sodium butyrate. But what we have seen here is that okay, the other thing, we had a positive control, which is azithromycin, AZT, which is one of the drugs that is used in the antiretroviral therapy. So we use this drug as a positive control. So in our screens, what we have seen is that the cultures where we had media only, there was no activity, and also where we spiked the media with these epigenetic modifiers, we had no activity. So now we are confident that whatever that we found is the fungi will be responsible. So then in our screens, we found that out of all these screens that we have, the valproic acid treated, we did not get any significant activity in the valproic acid treated, though this one, which is highlighted PO3, PL3 treated fungal culture, we had some activity, but the problem is that the IC50 was high, was low, and then indicated by this high number is supposed to be about 10 microgram per mil or lower to be significant. So there is no activity, although the graph showed that there is activity. But with the sodium butyrate treated cultures, what was interesting is that the PO3MP2 culture, we did see an IC50 of 9.3, which was very interesting. And then we said, yes, we are taking this culture as a yes, we are taking this culture forward. So we went on to identify these isolates. So this isolate identified as Penicillium chrysogenum, which is good because I have experience with working with Penicillium chrysogenum. And then we can also, if we want to sequence these, the full genome sequence, we have a reference strain which have been explored in natural products. So this was a good opportunity for us. So in terms of chemical characterization of our secondary metabolites, we follow this scheme which we adapted from Stockso et al. 2020, who identified a good compound with good activity against the HIV. So we said, okay, this is the strategy that they have followed that as adapted to our lab as well. So you have your crude extract, and then you do subfractionation. So in their efforts, they use a number of cartridges with mixed mode cartridges, which target different compounds, your anionic compound, the cationic compound, and also the hydrophilic, hydrophobic compounds, right? So they used 3 sets of cartridges to do the fractionation. And then after each fractionation step, you do the assay, which is a luciferase assay, and then we've termed this a bioassay guided isolation. So after doing the assay, we check if there is inhibition. If there is no inhibition, we throw away that sample, or we don't continue with it. We will only investigate other activities. But if there is inhibition, but we find that when we do the GCMS, the profile is too complex, then we send it back to SPE again, we fractionate again. But if there's sufficient purity, then we try to purify the compounds. So that's the stage where we are at. We are waiting for the results of our purification for the sample that we established it had sufficient purity. So let me conclude what we have done so far, or what we have achieved so far. So in our initial screens, we have shown that yes, indeed, endophytic fungi has the potential to produce anti-HIV compounds. And some of these compounds are encoded by the silent biosynthetic gene clusters, but we're also able to observe a shift in metabolite production after epigenetic induction using sodium butyrate. So what we need to know now is which genes are overexpressed. So that's one of the techniques that we can use to elucidate that is RNA-seq, which I'm currently trying to get more, I'm currently trying to get help from the University of Groningen in the Netherlands, because they've done the RNA-seq to check the differential expression of these fungal biosynthetic gene clusters. So one of the PhD there is helping us to establish this project, where we will look at which genes are more expressed, leading to the shift in metabolite that we see after treatment with sodium butyrate. So the other project that we have a student, a master's student, who is registered at the University of KwaZulu-Natal. She's taking this project further. She's developing various assays for mode of action. One of the mode of action that she's interested in is the immunomodulation. If these compounds that we have play a role in immunomodulation. So we are interested in the finding of that study, because it will expand our antiviral testing platform. So in our case, at University of Johannesburg, where I'm based now, at this point, I'm more interested in functional genomics, where we're looking at CRISPR-Cas-based genome editing to link the phenotype to the producing biosynthetic gene clusters. So what happens in this, you get a good full genome sequence, which we are still trying to sort out. And then from this full genome sequence, then you will identify, use different tools to identify the responsible biosynthetic gene clusters. And then once you've identified the biosynthetic gene cluster, you can do all these techniques, like your silencing, your overexpression, to see what happens, and to really study the regulation of the particular biosynthetic gene cluster. So I would like to thank you all for listening to my presentation today, and also Professor Deparati for inviting me to this talk. And first and then most importantly, I'd like to thank people I work with from our students, Bruce Nzimande, John Makutine, Avela Matandela, and Asipe Fanele, who have been active in this research. And I'd also like to thank my collaborators and contributors to this project, which is Dr. Nompumelelo Mkhwanazi, who's a virologist who help us with viral assays, Dr. Hezekiel Kumalo and Dr. Ndumiso Mhlongo, who have been my collaborators in the drug protein interaction, who are computational chemists. We do a lot of interesting work with them as well. Professor Arnold Dressen, I cannot thank this Professor Dressen enough. He have opened his lab for us to come and play and come and ask any question, and then he's kind of like mentoring us on the fungal secondary metabolites, especially the CRISPR based techniques. So the research is funded by the NRF, as I've said, I've been introduced, and I've also like to thank the HPP. They are doing interesting work. They've also allowed fungi in their lab. Most people don't want fungi in their lab, so they allowed fungal samples to be tested in their lab. We also would like to thank the PRF for funding us for the viral project, the universities that are hosting us. We'd like to thank them, and also the University of Groningen. Thank you all.
Yeah. Hello. Yeah. Yeah. It was a very interesting, and it's a novel talk, Dr. Shizrin Lolu. We are really very happy to hear. You started with fungal antimicrobial resistance, and especially when it comes to our earlier pandemic situation, or maybe forthcoming pandemic situations, especially those people who are affected with HIV. We know immunity is the main shield which has protected many people, and those people who are affected with HIV, because we know they are non-immune because of their health conditions. We don't know how badly they are affected during this pandemic, or during maybe forthcoming pandemics also. So your research, which has thrown light on how to activate the silent biosynthetic gene clusters with regard to HIV especially, you have done a really wonderful research, and we know, yes, we can understand how much pain you might have taken, and you cannot reveal your results before publishing them. But still, there are certain cases where actually with regard to CRISPR applications in filamentous fungi, where you are working with the latest technology, and really we are looking forward for a very good results from your lab. And regarding this lecture, I introduce another research scholar, Dr. Ajay Kumar, to take up the questionnaire. Thank you, Shizwe. But before that, I request my friend and Dr. Anjaneyulu to share appreciation certificate to Dr. Shizwe. Please accept it, sir.
Thank you. I accept the appreciation.
Maybe the next slide. The wrong slide.
Next slide.
Yeah, please.
Yes. Thank you so much. I accept it.
Our pleasure, sir. Please. Over to Ajay. Yeah. Yeah, Ajay.
Continue.
Unmute. Unmute, ma'am.
Ajay, please unmute yourself. Unmute.
Ajay, unmute your audio, ma'am. Yeah.
Hello.
Yeah, no, yeah, right.
Okay. Actually, good afternoon, sir.
Yeah, please.
Afternoon.
It is very interesting topic, sir. It is very useful to us and worldwide also. Actually, my question is, how sodium butyrate functions in epigenetic induction?
So, hello. Repeat your question. Sir, his question is.
Hello.
Sir, I think his question is how sodium bitrate functions in epigenetic induction.
Oh, so sodium butyrate is an HDAC inhibitor, so it inhibits the histone deacetylase. So once the histone deacetylase is inhibited, then it opens up the fungi from a euchromatin, from a heterochromatin state to a euchromatin.
Okay. Yeah. So
okay, sir. Thank you, sir.
Yes, sir. From my side, sir, do epigenetic changes, that is normally we go for methylation or demethylation, acetylation or deacetylation and all, no? Do they affect chromatin remodeling?
Can you repeat your question?
So do epigenetic changes affect chromatin remodeling? Because we do methylation or acetylation or demethylation, deacetylation, no, for during this process. Do they affect chromatin remodeling?
Yes, basically the chromatin, it's highly compacted, right? So if you use these compounds that are inhibitors of the histone deacetylases or inhibitors of other post-translational modifications, what they do is that depending on the compounds, as I've said, that's it differential expression. Some of the compounds, when you introduce them to the chromatin, they can further repress the chromatin. Some of the compounds, they can open up the chromatin, right? So it depends on the post-translational modification that you are looking at, especially DNA methylation. The output is not always overexpression. Sometimes it's repression. And you can also look at cancer intervention, where they've used these epigenetic drugs to try and cure cancer. Also in HIV, when they try to when they use these drugs as latency reversing agents. So basically, they will activate the latent HIV, and once it's activated, then the CD4/8 will attack the activated HIV. So yes, my answer to your question, yes, it alters the chromatin state.
Sir, one last question, sir, because I know again another speaker is waiting, and it's almost we are left with 45 minutes. How safe are the drugs? Because you are working with the fungi for producing anti-retroviral drugs. How safe are they when it comes to usage for humans, sir?
Yeah, I did not show you the cytotoxicity, right? So we in our initial screen, before we do the antiviral screening, we do cytotoxicity testing, right, on different cells. So what we have shown in the cytotoxicity cells is that they are not cytotoxic, right? But remember, these crude extract mostly that we have worked with. So we will see when we have pure compounds how they how the toxicity looks. And if you remember that in drug development, no matter what is your activity of your drug, if it's cytotoxic, then you cannot develop it further.
So hope your lab will produce really a good novel drug towards this HIV, sir, because almost 2 decades over, we are still waiting for a very good drug that can cure this disease. So we appreciate your research and your team, sir. So and even so heartfelt thanks for you for sparing your very valuable time and giving us a lecture here. Thank you, sir. Thank you.
Thanks, sir.
Now I request my other colleague, Dr. Shanti Priya, to introduce the last speaker, Dr. Devati Paul.
Thank you, sir. Dr. Devati Paul, currently working as Associate Professor in Amity Institute of Biotechnology, Amity University, Noida. She has completed her PhD from Institute of Microbial Technology, IMTech CSIR, Chandigarh, and postdoc from AWOG, Switzerland, and Mississippi State University, USA, for about 5 years before joining Amity University, UP, as assistant professor. Her main area of research is genomics, environmental biotechnology, antimicrobial resistance, and biodegradation. She has published more than 70 publications, including research articles, reviews, conference proceedings, and book chapters. She has received the prestigious Indo-US Science and Technology Forum Award, DST travel grants, etc. She also trains the students for skill development in quality control, chemist, microbiology, since from 2019. With this brief introduction, I welcome Dr. Dibattipal, and I request you, ma'am, please deliver your talk. Thank you.
Thank you so much for such a nice, I'm really floored. I'll just share my slides. Let me know if they are visible. Are they visible on full screen mode? Are the slides visible on full screen? Yeah, and okay. And I'm audible properly?
Yeah, ma'am, please continue.
Okay, thank you so much. I'll just put off the video so that there is no problem. So, as already Dr. Shanti Priya told, that today I will be talking on oleaginous red yeast strains for producing single cell oil and carotenoid, and it is a waste to value approach. So all along what my research is based on how to convert all the waste so that we have valuable end products. Currently I am working in Amity University and I am an associate professor there. So very briefly about myself, I have done my PhD from the Institute of Microbial Technology in a CSIR lab, and there my main topic for research was molecular and biochemical studies on biodegradation of paranitrophenol and 4-nitrocatechol, and also on bacterial chemotaxis. Then after that, I went abroad to Switzerland and also to Mississippi State University, where I actually studied how to work with yeast, and especially oleaginous yeast. And from there itself, I started working with oleaginous yeast, not only for biofuel production, also for the production of other valuable products. And thereafter, I went to Gainesville, Florida, where I studied about microbial molecular ecology. So recently, I am a faculty associate professor at Amity Institute of Biotechnology, and here my research interests are based to value-added commodities, biofuels from oleaginous yeast, and also bioremediation. Now, what is oleaginous yeast? We all know that nowadays people are laying a lot of stress to understand and study about getting biofuels from microbes. So oleaginous yeast strains actually accumulate a lot of lipid, above 40 to 50% of lipid with respect to their biomass. And the examples are Rhodosporidium species, Yarovia species, etc. So some of them also produce a red pigment called carotenoids. And this is the product which I was interested in. So most of these oleaginous yeast strains are non-pathogenic and they are easy to grow and easy to harvest, unlike many other carotenoid producing algal strains, which are again easy to grow, but it is difficult to harvest the product from the algae because of the difficult downstreaming process. Also, in the presence of many carbon-rich and nitrogen-limited condition, these oleaginous yeast, they can produce lipid droplets and store it within their cellular bodies. And in carbon-limited conditions, the same red yeast can produce the carotenoid pigment. So just by altering the condition of culturing, we can get two very important products from the same yeast. And most importantly, these oleaginous species, they show the ability to metabolize a wide range of carbon compounds. It can be hexoses, it can be pentoses, even non-sugar carbon like acetate, and then sugar alcohols, etc. So because of their potential to grow on such a wide range of carbon, we can easily use them for growing on all kind of waste, which actually contains a lot of different type of sugars. So hydrolysis of lignocellulosic biomass, that is the food waste we can have agricultural waste, or from the forest we can have forestry waste. So all of that, if we hydrolyze it, we get a range of sugars, and only these oleaginous yeast strains are such that they can utilize almost all types of sugars which are present in the hydrolysates. So you see that previously also people have worked on different type of waste hydrolysates to grow these oleaginous yeast, like effluent from fish processing way, some starch, wastewater. So throughout the globe, people have tried to identify the different type of waste which are there in their area, and they have tried to use them as low-cost substrates for fermentation of oleaginous yeast. So looking at this, I identified that around our NCR region, the most important and very easily available waste, which is actually available in over tons, say 100 to 125 tons daily. It is available just in one Azadpur Mandi, that is in New Delhi. So much of waste, it is simply kept on the streets or in that area where the mandi is. Mandi, to most of the my friends, I think they would identify the word as roadside vegetable markets. So this is the area and this is the waste that we identified for utilizing as the low cost substrates for fermentation of our yeast. And in that time, the Times of India, the newspaper, it said that there was an unbearable stench which was coming out of rotting vegetables, because the vegetables and the parts which people didn't take home, it was all lying there and creating a lot of disturbance, and it was also becoming a site for pathogens, rodents, and so therefore we thought that yes, this is the right thing that we can take from here. There is a lot of sugar which is just going waste and we can still use it. The only thing that the municipality did was to, at times already the rotten vegetables used to be taken to the landfill, but that is not a very good way of dealing with the waste, because you can make biocompost definitely, but still all that extra sugar, it cannot be utilized completely. The sugar can be used by the yeast, and then the remaining part, which is the non-sugar part, that might be used for the biocompost. So from there it started that I from waste to wealth research, that became my main motive. So the microbial lipids, they are already promising alternatives to the traditional plant and animal-based oils, which are used in so many different aspects in our life. And these microbial lipids, they also have a very important thing, which is the triacyl glycerides, and they are very important for the production of biodiesel, lubricants, surfactants, and oleochemicals. Now, these lipids are comprised of both saturated and unsaturated fatty acids, and we will discuss about it a little later in the talk. And also the carotenoids, which are produced by this yeast, they have three major fractions that we found: beta carotene, tauruline, and taurular rhodin, and all of these are antioxidants. They have anti-neoplastic character, and I will also tell you that how we found many more different purposes for which we can use the carotenoid. So first thing what we did, we have to derive a very suitable fermentation media from the mandi waste that we accumulated, and that will be the feedstock for cultivating the oleaginous yeast strain. Then we isolated many different oleaginous yeast strains for the production of microbial lipid and carotenoid pigment. And then we developed a process that we started from a batch, and then we went to the fed batch fermentation, and how we have increased the production to the maximum. So for the very first objective, we isolated oleaginous red yeast strains. We found that it has a lot of different type of carbon sources, xylose, glucose, and many others. Then we studied the colony morphology. We detected whether lipid globules are present or not using Sudan black staining. And then further we identified the strains using biochemical and phylogenetic analysis. So here are the results. You can see that we got different type of strains, and then when we stained with the help of Sudan black, all of them had some oil globules. So all the data we have collected that, and we have also identified the strains, and we got the strains submitted, the 18S that we have done, that are all submitted to the gene bank, and we have the accession numbers. So how we prepared for the fermentation medium, we first took the inedible waste material that we collected from the mandi, and we have first segregated the waste. That is important because first we have to identify how much of carbon and how much of nitrogen is contributed by each different type of waste. And once we have that, then later on we can use a mixed waste, and then just break it down to pieces, and we simply boiled them and autoclaved it so that we can collect all the sugar in the water that you can see. So this is simple water that we have taken, nothing like a distilled water, because we want to retain the mineral salts. So this medium that was formed, it is a complete medium. There is nothing that it will take a lot of money to make this medium, and we do not have to add anything extra to it, like we do not have to add any special nitrogen or carbon source or any mineral salts, etc. In itself, this was a very complete medium. We did the nutritional analysis of this medium, and we also did HPLC analysis for determining the different type of sugars which are there. So we can see a lot of different types of sugars, and also apart from the carbon and nitrogen, we found that 0.18% phosphorus and 0.56% potassium were present in the waste extract. So with this medium in hand, we then went ahead. See, these are the different type of sugars. Xylohexose, glucose, that is the maximum. Xylose is also in a lot of amount. Glycerol was less, but at different type of conditions, we get sometimes more or sometimes less of glycerol. But in any case, glucose and xylose was mostly present in the medium. Now, when we grew this organism in this medium, we found that, very interestingly, this organism utilized three different, that is glucose, then xylose, and then glycerol, and all of them, while they are using it, they showed a trioxic growth pattern. That means that the log phase, as you can see, was there 3 times, corresponding with the glucose, xylose, and glycerol. So you can see that once the glucose is first nearly consumed. Then in the second step, the xylose is preferentially consumed. And after that, glycerol is consumed. But the glycerol, when it is consumed at a very fast speed. And during this stage, in this graph, we can see that not only that the biomass, but lipid production is also the maximum when the glycerol is also being utilized. So you can say that in the later stages of the log phase, we have glucose, we have lipid production. And toward the end, that is after the stationary phase is nearly over, we have the carotenoid production. So this is the comparison of lipid produced and the biomass in all the strains that we had isolated, and also 2 of the strains that we have procured from the different gene banks. So here we see that this strain ATCC, it gave the maximum one, and followed by strain AB1. But although in this we can see that the lipid production is around 13.4 gram per liter, and productivity is also quite high, we also see that the organism has been able to store around 73 or 74% of lipid as compared to the biomass. But still, we wanted to increase the lipid production a little more, because 13.4 gram per liter is not a very, it can still be challenged, and we felt that we can increase the production a little more. So for that, we cannot force the organism to have more lipid content. But what we thought we can do is we can increase the carbon so that more amount of lipid or more amount of biomass is produced. Just excuse me 1 minute. So what we wanted to do is we just increased the carbon. Now to increase carbon, that means in the waste extract we have to supplement something. So we tried to see what can be the other kind of possible cheap or easily available source of sugar, and we found that sugarcane molasses can be used. So then we tried to fortify the medium with sugarcane molasses, and we found that the lipid production was a little better, although it was not enhanced to the fullest. So at this stage itself, we started comparing our data with the existing literature so that we can understand where we stand and how much better is the scope. So at this time, we found that yes, the waste extract was a very good medium, and even if we do not fortify it with other carbon sources, we are still getting a very good lipid content, which the other reports did not say that the yeast can grow so well on the other kind of waste hydrolysates. But we also wanted that our data should be like comparable to that which is being used in synthetic medium. And in synthetic medium, definitely the lipid content would have been better. Then we also tried to see that if we transesterified the lipid that we have already obtained, then how does it look? So we did the TLC analysis after we trans-esterified it, and we found that it has lots of tags, DAGs, and MAGs, that is the triacyl glycerol, the diacyl glycerol, and monoacyl glycerols. And the highest amount of tag was determined in ATCC strain. So we decided to go on with this strain because tag is the one which is the most important type of lipid, or the triacyl glycerols is the most important type of lipid that is lucrative, and so we wanted to go ahead with this strain. But all the other strains also produced quite a lot, or I mean, if you compare with soya oil, then we can see that the amount of tag in the microbes are also very, very promising. Then we went ahead for preparing biodiesel by again trans-esterification, and we have done GCMS analysis. We found that in ATCC strain, the saturated and unsaturated fatty acid proportions were also very promising. We have published all of this data. The kind of biodiesel that we were getting by using acid trans-esterification, we tried a different number of different types of trans-esterification, but this method gave us the best results. And we can see the type of biodiesel that we obtained. So from the extracted lipid, we can see that we got around 10.5 gram of biodiesel. So you can say that just one fermenter run can give us this kind of this amount of biodiesel. So this was quite challenging, and it looked good. So we tried to now see whether the biodiesel that we have obtained is according to what the ASTM, or the American Society for Testing and Materials, if their whatever clauses they have for the biodiesel, if our data is also matching the same or not. And it did match in most of the cases. And wherever there are differences, we found that, yeah, in microbial oil, there will be some differences as compared to plant oil and or animal fat oil. But this also suggested that some type of process optimization might be done still to make it a better product. After this, the challenge was also that we want to increase the amount of lipid, because if we increase the amount of lipid, the amount of products also will increase. That means from lipid, whatever we can make, we can either make biodiesel, or we can use the unsaturated part of the microbial lipid, and we can it is as good as salmon oil or cod liver oil. So that is the healthy part of oil, and we can use it for fortifying our normal oils that we use as a cooking oil. So so right now the main object was to increase the amount of biomass, and therefore to increase the amount of lipid that we could get by 1 or 2 fermenter runs. That is a 10-liter fermenter. If we ran once, then how much we can increase, and then that will also help us to scale up the process. So one very important thing that we realized at this point was, if we gave the organism some stress, that is stressed it with some osmotic shock, that is by adding NaCl, then the organism seemed to increase the amount of the lipid that it formed. But this strategy, although it was a good strategy, the productivity that we wanted could not get reached. So that means we were not in the end of our search. We needed to go ahead. So then we decided for fed batch fermentation. And for doing fed batch fermentation, we have to see that where is the substrate inhibition point. That is, if we fortify it with glucose, then we should know that how much of glucose is safely to be added to the medium, and after what point the organism can no more take the that much of glucose, and the substrate in itself will inhibit the fermentation process. So for that reason, we did this kind of study. We found that the glucose at 90 gram per liter, that is quite a high amount of glucose, that is easy. The organism can still take it, but after that, the amount of glucose starts to inhibit the substrate, the product formation. Then we again thought that glucose, if we try to fortify or make the fermentation medium fortify with glucose, it is going to add to the cost. So we do not want to add to any of the cost. So that is the reason we again went back to sugarcane molasses, which we had tested before, and we found that it is giving a good growth. So at 30 gram per liter, sugarcane molasses was giving quite a good biomass, and the lipid production was also good. So then we went to fed batch fermentation, and we started feeding the fermentation media as soon as we found that the glucose level went below 20 gram per liter. So as soon as we did not have a fermentation, fed batch fermentation, that we are going to add the feed at particular or fixed time points, but we always try to feed the medium whenever the sugar fell below 20 gram per liter. Now this is very important in this step, because this organism not only utilizes the glucose, but it also utilizes many other types of carbons, as I told before. So until and unless the total sugar was below a certain point, and we feed, if we try to feed it, then again substrate inhibition occurs. So we had to be careful at this point, and we have fed both the strains, the ATCC species and the AB1 species, with glucose and also with molasses, and these were the results obtained. So finally, what we got was that the lipid quantity that we wanted was 39.2, starting from the 13 gram per liter. So we can say that we were happy with our results, and we have got such a good amount of production of lipid. And these results also we have published in 2020 in Fuel. So ultimately, we can say that this is the nutshell of what we have done. At first, we have gone for isolating the different type of yeast. Then we have prepared a good medium with the help of waste. Then we went ahead for batch fermentation in the presence of glucose. sugarcane molasses, and also by fortifying with NaCl, that is for osmotic stress. Then we have got the lipid, and we trans-esterified with the help of fly and dyer method. And the biodiesel we have characterized, and we found that it is suitable for use in many of the automobiles, and they did stand by the ASTM criteria. Also, we found that lot of other type of or the other yeast which have been grown in different type of fermenters by different methods, the lipid content that they have got and what we have got is a little higher. So we were actually happy with our own work, and we didn't stop at it. We now went ahead to see that how best we can use the lipids so that we are able to get different type of products. The other product that we I didn't talk about in the fermentation is the carotenoids, and it has its own story, because of course it will be again obtained through fermentation, but this fermentation for carotenoid production is a little different. And in the case of lipids, where I said that we are fortifying it with molasses or we are fortifying with glucose, here we have to see that the carbon remains low. So then only we are able to get a good amount of carotenoid pigment. The other challenge was that the hydrolysates of waste, many times they have some very a high quantity of inhibitors. So we had to dilute the medium, with minimal medium, and we came across different type of lignocellulosics with which we have made hydrolysate, and then with the dilute one which had very less carbon, we have been able to still do the fermentation and get carotenoid pigment, which we have then extracted. Now these carotenoid pigments, they are the precursors of vitamin A. So definitely they are they have a lot of antioxidant function like all the vitamins, and we have found that they have utilization in cardiovascular diseases, in cancer and ophthalmological diseases. And of course, now we have added one more to this basket. That is, we found that they have anti-malarial activity, and we have published this result very recently. We also found that this anti-malarial, along with the anti-malarial activity, they are very good fighters against breast cancer. So for females, I would say a lot of carotenoid, if taken through the diet, might be very useful for preventing breast cancer and many other diseases. So in a nutshell, I would conclude today that we found that our mandi waste is a very rich amalgamation of various carbon sources containing many essential minerals required for the growth of microbes, and therefore it serves as a complete fermentation medium. Oleaginous red yeast strains were isolated from various sources and identified and then used for lipid and carotenoid production. All oleaginous yeast strains utilized various sugars that were present in the waste extract and consequently accumulated high amounts of lipids and carotenoids. And fed batch fermentation enhanced the lipid production, and the physicochemical properties of biodiesel reported in the study were within the range of ASTM standards. So these were some of the publications that we could make with the help of this work. And I really thank all of you for my patient listening. And before I conclude, I would really like to thank our founder president, Dr. Ashok K. Chauhan, for all the necessary support and facilities that he has given to us, the head of our institute, the ex-head, Professor Chandandeep Tandon, the center heads, A.N. Sahi sir, and my various colleagues who have helped me throughout the journey, especially Dr. Christine Jaisilian and Dr. Anju Arora. And of course, I cannot forget Professor Mark Lawrence from Mississippi State University, and my dear students, Gunjan, Shweta, Chetna, Eksimar, and all the lab technicians and attendants for their hard work and dedication. And my special thanks today is for the faculty of Palamuru University and IQAC team, and the esteemed principal, Madam Registrar, Palamuru University, and Vice Chancellor, Palamuru University. Heartfelt thanks to all of you, and also to the other speakers, because they have really made my day by joining in from such far away places, and really I appreciate all of it. Thank you so much.
Yeah. So thanks a lot, Dr. Debati Paul. Really, it was a wonderful session.
Thank you.
You have given really a good solution for dealing with the vegetable waste to a useful product. So before starting any questions from the Department of Microbiology, like from our senior Professor Pindi Pawan Kumar sir, who is Registrar and Chairman Board of Studies, and myself and my good friend and colleague Dr. N. Kishore, Head Department of Microbiology, and Dr. A. Shantipriya, Dr. S. Mudula, and Dr. B. Sridevi from campus, and Dr. Shankar and Sanjeev from Peace Center Vanapati, our non-teaching staff, and even our lab assistants, Revati and Ramesh. We all really extend heartfelt thanks to you, because you are the key person behind this for arranging your friends for this webinar. So thanks a lot for your support. And I will ask my another research scholar, Jyoti, to handle the question hour session. Over to Jyoti, please. And before that, I request Dr. Debati Paul to accept our appreciation certificate, please.
Thank you so much for this appreciation. It means a lot to me. Maybe.
One second. Yeah, it's coming. 1 second. Can you able to say? I think, yeah. Is it visible, ma'am?
Yeah, I can see the pink slide, but I think the second slide has to be focused.
Because
Okay.
Yeah. Yeah, please.
Thank you so much.
Yeah.
Yeah, thanks a lot.
I'll send it.
I can understand during webinars we can understand there are some glitches, but apart from all the glitches, I think the facility that we are able to hear at one point of time from across the globe, that is a very good thing.
No, your certificate was coming with more acknowledgement. That way it took time, actually.
Okay. That is the positivity which I always get.
Yeah. Thank you, ma'am. Yeah. So over to Jyoti, ma'am, please. Jyoti, please, I unmute you. Yeah, please. Yeah.
So good afternoon to all of you. Good afternoon to ma'am, Debati ma'am, and also dignitaries present in the webinar today. So hope I'm audible to all of you.
Yeah, please.
Thank you for giving me this opportunity. So I'm Jyoti, research scholar from the Department of Microbiology, Palamuri University. So ma'am, your lecture has given us great insights on production of biodiesels from oleaginous yeast. It has given us a clear picture and also explains of exploiting the beneficial aspects of our own invisible world for the benefit of humankind. It also has given information for research scholars and also for the students of converting waste into wealth. And it also gives us mainly innovative ideas for new startups also in the field of industrial microbiology. Thank you, ma'am. So your lecture was really very, very informative and simple to follow. So from my end, I just have a small question, ma'am. So, and then I will mainly move ahead to the question answer session. So I would just like to ask you, so what are the major natural habitats from where we can isolate these oleaginous yeast, ma'am?
Very easily you can isolate from all these flower petals. Wherever you think the sugars will be there in the nectar of the petals, on the skins of some fruits you will get. So I use different type of peels, petals, and we also used some soil also. For soil we didn't get so many good results. I got more good results from peels of the fruits. The special thing that is very interesting about these organisms, that they are able to use pentoses and hexoses equally well. This characteristic is not there in other type of organisms. So wherever you feel that there will be mixture of both type of sugars, there these organisms will grow.
Thank you so much, ma'am. And ma'am, also I just would like to ask you, so when we have designed the fermentation medium, that is the waste extract medium, so how do we eliminate the growth of other microbial strains, because it has rich sugar content and all? So how do we make it selective only for our oleaginous strains?
Yes, that's a very good question. Number one, we found that this strain is robust. It did not allow the growth of bacteria at all. So we did not get that kind of contamination. And I did not have to use any kind of antibiotic or antifungal stuff in it. So I can say that maybe it is the feature of this yeast that it will prevent the growth of other kind of strains. But where in the fed batch fermentation, where we have to increase the amount of sugar, and at that time there was a problem. So we have used simple NaCl.
Okay, ma'am. Okay.
And at a low concentration, 1% to 5%, and that was enough.
Okay, thank you, ma'am. Ma'am, so comparatively, because we have seen that these species of Rhodopteryopsis and all, they have produced biodiesel. So are there also any comparative studies which you have done related to already known species which is producing biodiesel?
Yes, I showed that in the slide.
Especially with the bacterial strains, any bacterial strain which has produced, and you have
I
done
didn't compare to bacteria. I didn't compare to bacteria. We have published the results where we have compared to algae and plant and animal fats. Bacteria is difficult, not difficult, I would say. It is not a good source, I would say, because the bacterial cell in itself is so small. So the amount of lipid which it will accumulate will be also very small. And another thing is, this yeast is very like this system, that if you study the genomics also, we are able to manipulate the system very easily. And not only the lipid and carotenoids, now we will try to also get other kind of valuable products, like from sandalwood we get santoline. We can produce that same in this kind of yeast also. So that will be our next level of work that we are focusing, and we want to write grants to get some.
Thank you so much, ma'am.
Yeah.
So anyone who have any related to ma'am's topic, so you can please go ahead. So
yeah, Dr. Zeta Potential, I have a question for you. Hello.
Yes, I am able to hear you.
Yeah. So can we use this microbial lipids as edible oil or at least for blending?
Yes, we should actually use it for that instead of producing biodiesel, because for biodiesel already I think there are other methods which are more, I would say they are better and they are enhanced, and we can actually use this lipids for fortifying the cooking oil that we have. That will be better because it has lot of tags, and we can use this. If we just add these oils and fortify the cooking oil, it will become a better cooking oil.
Because what we heard recently is they are using oil hydrocarbon wastages, which is very dangerous to our health. Or even there are reports that they are extracting oil from bones and all, from dead animals.
Oh.
So instead of that, this would be better, though it is from food wastage, but it's a plant source.
Yes, it is a microbial source, completely biological source. And see, from the waste, some people ask me one question, that you are taking so much waste, the waste will also have pathogens. But we don't use the waste. We taken the hydrolyzed from the waste, and whatever was remaining from there, we have discarded and we have made biocompost from there. So this, we have just taken the hydrolyzed, that's the sugar part. So pathogens will not come.
If some toxins are released into the extract medium, do you think any toxins will be released into the extract medium and it might be harmful or something like that?
I'm sorry, I didn't. Please come again one more time, please.
No, what she means is like you are taking the hydrolyzed, no? So suppose if any toxin comes into that.
Okay, this is one more thing, that we made this method very simple. We are not adding any extra acid or any kind of alkali. So there will be no inhibitors in the entire thing. We are only under high pressure, we are steaming it. And steam is in industry, if we take this method to industry, steam is very much conveniently available in the industry. So we have to simply take normal water, the one we can get from our RO, and we just put our waste in it and steam it under pressure. So we will be able to get the sugars will come out into the medium.
Last question, ma'am, because I know it's already 2 o'clock. So almost 10 minutes we are, So last question is like, in your results, you have used glucose and molasses also, right?
Yeah.
So normally we say that glucose is a pure and easily available source, whereas molasses is
glucose we don't want to use because it is very expensive. Glucose we started to use just to try whether it will work or not work. When it was working, we took molasses.
But what we observed is the yield is less when compared to when you have used molasses.
Molasses, actually it has more minerals. There are other things in molasses which has helped in the growth. That's what we saw.
Okay.
Because of the other nutritional capabilities of molasses.
Okay. So now so really it was a very good session, ma'am. I request another colleague, Dr. Shanti Priya, to propose vote of thanks for this webinar. Last but not the least, once again I thank all the expertise pioneer for delivering their lectures. Over to Dr. Shanti Priya, please.
Thank you so much.
Good afternoon, everyone. Respected chief guest and chief patron of this event, Honorable Vice Chancellor, Sir, Professor L.B. Lakshmikant Rathod Garu, and Chairman and Registrar, Professor Pindi Pavan Kumar, Sir, Principal, PG College, Dr. Noorjahan Ma'am, IQAC Director, Dr. Madhusudan Reddy, Sir, HOD, Department of Microbiology, Dr. N. Kishore, Sir, and my colleague friends and all my dear participants. I feel it's a great privilege to propose vote of thanks, and it gave me immense joy to be the co-convener of this webinar, New Frontiers in Biotechnology Research for Social and Environmental Welfare. On behalf of the Department of Microbiology, Palamuru University, I would like to thank our Honorable Vice Chancellor, Sir, and respected Registrar, Sir, for their continuous support and encouragement. I especially thank our speakers, Dr. Suma Mukherjee, ma'am, Dr. Chijwe, and Dr. Dipatipal, ma'am, for accepting our invitation and for delivering valuable lectures. We are thankful to you all. I would also like to express my sincere gratitude to all the participants, including professors, faculty members, research scholars, and students who have participated in this event and making this webinar a success. Not last for the least, I extend my heartfelt thanks to the technical team, electrician, and media for their support. I hope all of us have gained good knowledge from this webinar. Looking forward to conduct such many events in the future, which can be helpful for the microbiology fraternity. Thank you, one and all. Thank you.
Thank you.